You have, but why? Scientists said, why is it that these traits are inherited? How does it get inherited? What is it that is inherited? Yeah.
So then, wouldn't you be looking at the phenotype of it then?
Yeah, you're looking at the phenotype. People looked at phenotypes when they did this. They looked at phenotypes. And early on, people thought that the males carried everything that you needed to carry in order to have a baby. It was the female was just the soil upon which males planted their seed. Females were just where our babies grew. We, in our sperm, was everything you needed. Right? This is called the homunculus. I can't pronounce it. But the thought was that everything that is a human, inside a human, is like a seed. The sperm is like a seed. What is a seed? Who knows what a seed is? When I say seed, what's the image that's in your brain?
Plant like sunflowers.
All right, take a sunflower seed and you plant it, and then what happens? It grows. So that seed has everything you need for that sunflower to grow. Is that correct?
No, it doesn't have the soil.
Okay, but yes, it needs nutrients and soil and other. That's a good point. But it's already a sunflower. It just has to what? Grow. Is that correct? That's your understanding of a seed. That's what people thought of when they thought of sperm. Sperm were seeds. But they're not seeds. They're only half a seed. They only have a seed. Because a seed needs two parts, and we'll talk more about that. That's just kind of an illusion. So they were wrong. That's not how it works. So, as you saw from the experiments that you looked at, Griffith was looking at in 1920. Now this is 1920s. These thoughts were happening in 1600s, 1700s, right? Think of Mendel, right? Think of Isaac Newton. Those guys. They all thought that we all sow seeds, and the woman is like the fertile soil upon which our seeds grow, and that's it, right? So that's how it goes. Well, here comes 1920s, and this gentleman. And by then, we pretty much were sure that's not what was going on. Because we understood already that you need a sperm and an egg to make a seed, a single creature, right? A new creature. And we knew it was a new creature by then. We knew the children weren't exactly like the parents. They were slightly different, but yet they carried something. They inherited something from the parent. So the question was, what is it they inherited? Well, this scientist was working with S. pneumoniae. What is S. pneumoniae? What is it? What is it? A strain of what? Of pneumonia. What type of pneumonia? Viral or bacterial? Bacterial. There's two types of pneumonia at least. There's bacterial and there's viral. This is a bacterial pneumonia. So this is a prokaryote. Is that correct? Okay, as long as we're all clear on that. So it turns out that prokaryotes, and specifically pneumonia, has this ability to produce a mucus layer. The mucus layer hides the proteins on the surface of the cell and protects the bacteria from the immune system. The immune system cannot recognize the bacteria if it can't see the proteins that are on the surface of the cell. So if it's smooth, a smooth strain, right, then what happens? Then the immune system can't attack it, and what does the bacteria do? It causes death, disease and death, right? Because the disease-causing bacteria, the body of in this case, the mouse or the rat, in your case, the human, can't detect it, can't destroy it. The identification process that this is an invader only happens if the proteins, the cell surface proteins, are visible, right? And they're invisible with that mucus layer. So we knew there were two strains. There was a rough without the mucus layer, and then the smooth with the mucus layer. And you can see them under a microscope. If I'm going to go ahead and flip back over to the computer screen so you can see it yourself. And you have it in front of you in your handout. And you can see that the rough strain looks like this, and the smooth strain actually looks like little droplets, almost like droplets of water, right? All right. So you had basically 4 categories. You had a rough strain, which we know does not cause death, right? And we know that the smooth strain does cause death. And we know that we can kill it, which means if it's heat killed, what does it mean? It's dead, right? If you inject that into the mouse lives, right? So the rough, it lives. If it's smooth, it dies. If I take the smooth and I heat kill it, so this is heat killed smooth, and I kill the smooth strain, it lives. What is this? What is this group here called? And everyone should know this. What's this heat killed? What do you call that in this? What group is that? Control group. Excellent. Why is it a control group? What are you looking for? Is it a negative or positive control? It's a negative control, that's right. This is your negative control. Why is it negative control? Because you don't expect it to die. Because if this thing lives, then you know that it's not, what are you looking for? Here's the question they're answering with this. Or you go ahead and tell me, what question are they answering with this?
They're answering the question of, what if the mouse died for some other reason?
That's exactly right. What if there's something about the bacteria that's not the bacteria? What if it's not the bacteria that's killing the mouse? What if it's just something that's on the bacteria? Or what's something that maybe it's a bacteria poop, or maybe it's a bacteria, something that the bacteria produces is a toxin that the bacteria produces, not the bacteria itself, right? So it's not inherited, it's just it's some other factor that we don't know. Like for instance, did you know botulism? You've heard of botulism? It's a food poisoning that kills you. It's actually a toxin. It's not the bacteria, botulism bacteria, that causes the death. It's the toxin that the bacteria produces. So you can cook botulism and still die. Because you'll kill the bacteria, but you'll still die because the poison's still in the food. So it's interesting that's what they were testing, to see if it's the bacteria or something else that's killing the bacteria. So then they did one other experiment, there was one other group that was interesting. So they knew that the rough strain, these are controls. These are positive control. These are positive controls, or negative. This is negative control, positive control, negative control. Here they know the rough strain. You can be exposed to rough strain, they live. Here they know the smooth strain kills you. And here they know that the heat killed smooth, you still live. So these 3 are your control groups. What's your experimental group? The one that you're interested in? Can the rough strain inherit the DNA, the something from the smooth strain that will kill? They'll make it smooth and kill. So you take a rough strain and you combine it with heat kill. Now when you heat kill something, you broke up the cell. And there's DNA, proteins, RNA, carbohydrates, all floating in the test tube. Now bacteria are not like eukaryotes. They can take in DNA and use that DNA. They have things called plasmids. Their DNA is round, by the way. Ours is linear. As you know, ours has a 3' end and 5' end. And bacterial DNA is round. It's connected, it's a circle. And bacterial DNA can be absorbed by a bacteria. It can take in DNA and use it from other bacteria. That's how you get antibiotic-resistant bacteria, or at least one way. So the rough strain are heat killed, and they're combined. So the rough, and then heat killed smooth strain. So that means what? That you add these 2 together, and what happens? The mouse dies. What does that tell you?
The rough strain inherited something.
The rough strain inherited something that allowed the rough strain to become deadly. Something, and we don't know yet if it's DNA or RNA, right? But we know that it inherited something. Now we have these tools. We have these things called enzymes, and we've talked about enzymes. You've been tested on them, right? We know that they end with ACE, right? I hope you know that. So proteases, what do you think they do? Yeah? They break down proteins. So that if they break down proteins, that means the proteins aren't there anymore. So the question is, what is the stuff that bacteria inherited, right? Was it DNA? Was it protein? They were pretty sure that it was either protein, DNA, or RNA. They were pretty sure it was not a carbohydrate. They're pretty sure sugar was not what you inherited from your parents, right? They were pretty sure of that. Not 100% sure, but they were pretty sure. They actually thought for sure it was protein. I mean, think about it. Which makes more sense? If you're going to make something as complicated as you, right, with your incredible brain and your amazing talents and your emotions and your thinking and all the things that make you up in the society, not just you, but every living thing on this creature, right, all the different things we make, does it make more sense that the language that defines who you are has 20 letters? And what 20 letters am I talking about? Amino acids, right? Proteins. Or does it make more sense that you have 4 letters? And what 4 letters am I talking about there? Right, a DNA. So it's either DNA or it's protein. So most scientists honestly thought it was proteins. They were almost 100% sure it was going to be proteins. Because you had 20 letters, you can make all kinds of things. We knew that DNA was boring. 4 letters, and just repeated itself for a while. We didn't even know what DNA was. We thought DNA was just like, there's some intermediate, it's useless.
It's still on the packet.
Say what? Oh, I'm sorry. I should have switched over. My bad. All right, so proteases, most scientists thought it was proteins. Most scientists thought it was proteins. It was the inheritable material at this point. And it makes sense. And by the way, and this is repeated throughout history. The Egyptians, they did amazing chemistry. They would preserve human bodies. You know this, yes? They preserved human bodies for a long time. There's still some mummies around from thousands of years ago. Yeah.
Some of them still have hair.
They use preservatives like we have in our foods. Like we do with our own bodies when we bury our humans today. They have preservatives they use to preserve people. They wrap them. And one of the things they did is they took the organs out and they put the organs, the important organs, in jars. They called them Canopic Jars. So they'd take the heart, obviously very important. They would put it in a jar, preserve it. The liver, preserved it. But the brain, they thought it was useless. They took a hook, they stuck it up the nose and yanked it out. They thought it was mucus. It's like, throw that away. What is that? They never preserved the brain. See, throughout history, sometimes goes back to what I try to teach you in the very beginning of the year. We're never sure what's going on. We think we know. And we assume we know for now until we find out it doesn't work. So for thousands of years, the Egyptians thought the brain didn't do anything. They got rid of it when they were preserving their pharaohs' bodies. Yeah.
They left the heart inside of the body. They thought it was going to be the only thing necessary for the afterlife.
Okay. You learn something every day. All right, are we good? But what was the point? The point was what? That in humans... Oh, sorry, go ahead.
You can say it.
All right, so the point is that humans We think we know what's going on, but sometimes we're wrong. And here in this example, we thought proteins, 1920s, we thought proteins were the things that we inherited. When you got something from your mom and dad, it was some kind of protein that they got. All right, so the next thing is a DNase. What do you think DNase does? It breaks down DNA. And what does RNase do? No, they don't do it that way. So remember, that means breaks down. That means it's not going to be there if we break it down. So and this is true of most classic experiments. Most beautiful experiments are amazingly simple. The simpler the experiment, the clearer the results. The more complicated the experiments, the more possible. Every time you add a step, there's an extra chance that what? Something's going to go differently or wrong. So a simple experiment's usually the best. So they took this, what they did is they took the heat-killed, right? They took the heat-killed smooth bacteria. And I don't know why I'm doing this, because I already sent you the video on this, right? If you've done it, you've listened to it. But I think it's important that you understand that this is how we know that DNA is the inheritable material. So they added, to one test tube, they added RNase, right? What does that mean? They took the heat-killed smooth strain, they added RNase to it. What does that mean?
The RNA is going to be breaking down.
The RNA is gone. So what's left in there? So what's left in there besides carbohydrates and fats? Proteins are there, check, and DNA is there, check. Okay, they add that to the rough strain, and what happens? So what does that mean? It's not RNA, right? RNA is not the inheritable material, because if it was, then it wouldn't die, right? Because if you got rid of RNA, and RNA was the inheritable material, the rough strain would never inherit the ability to kill the mucus layer. Really simple, beautiful, straightforward, right? So that's experiment one. And what's the second experiment is they took the same heat kill, same stuff, everything else they kept the same, right? Because that's how we do experiments. And you took it in a test tube, and you added a protease. And what did that do? No protein, right? But what did you have? DNA was there, and what else was there? RNA was there. And what happened? Someone said it already. They added that to the rough strain, and what happened? You had death. So again, that proved what?
It's not a protein.
It's not a protein. So here we know it's not RNA, the inheritable material, not protein. And what's the last? What did we add to the last group? Was what? We added a DNase, which got rid of no DNA. But there was RNA, and there were proteins. When we got rid of the DNA and added that stuff to the rough strain, what happened? It lived. So what does that mean? And that's it. That was the experiment that showed that DNA was what was being inherited. We still didn't know what DNA was. We knew it was 4 different types, A, T, G, C. And there's a couple others, but at least A, T, G, C, right? And we didn't know how, anything else about it. It wasn't until Watson and Crick and Rosalind Franklin that we got an idea of what DNA was. So the next experiment was DNA replication. So here, with DNA replication, what do we know about DNA? What do you know about DNA? So give me some information you know about DNA. Yeah. It's double-stranded. So there's two molecules, two strands of DNA. 5' to 3', and 3' to 5'. Yeah?
It's shaped in a double helix.
It's a double helix. What else? Yeah?
It's monomers or nucleotides.
Nucleotides, that's right. A and T and G and C. God, you guys are making me feel better. All right, what else? Yeah?
It is held together by hydrogen bonds.
Say again? They're held together by hydrogen bonds. Excellent. These are hydrogen bonds holding these strands together. Okay, yeah?
It has a sugar phosphate backbone.
Sugar phosphate backbone. Excellent. Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. This is the backbone. Does everybody understand that? This is the backbone here. All right, yeah?
They are wrapped around histones in the nucleus.
That's right. They're wrapped around histones, and that's another story. I don't want to get into that right now, but yes, that's true. Yeah?
They have nitrogen spaces.
They have nitrogen spaces. That's these guys, right? Yeah?
It's missing an oxygen on 2'.
Missing an oxygen on the second carbon. That's right. Yeah?
It has, when you unravel it, it's about a yard long. Wait, is it
That's
yard?
what it is. It's 3 meters, I think, about 3 meters long. So you know what? A yard is fine. It's long, right? I mean, the exact length, who cares? It's long. It's really, really super long. It's a molecule, so that means a lot of nucleotides, because remember, the nucleotides are very small. So there's billions of nucleotides in each cell. So it's just an idea. It's just, the whole point is that there's a lot of information there. Right? Yeah?
Held together by covalent bonds.
The sugar phosphate backbone is held together by covalent bonds. The hydrogen bonds that hold the two strands together. Excellent. Excellent work, ladies and gentlemen. All right, so that's really a lot of information. A lot more than they had that Meselson and Stahl had. Meselson and Stahl didn't have as much information. You know more than they did then. You know more right now than these scientists did then. They did not understand what the heck was going on. They did know that there were copies being made. They did know copies were being made. Now we're in 1958. The previous experiment was 1920s, right? 30 years later, one generation or 2 generations later. 1958. How is DNA copied? How do we copy DNA? And that's the question. You have one strand, and we know you know this thing called mitosis and meiosis occurs, but you have this strand. Let's use 2 different colors if you don't mind, but we'll start with one color. You have 2 strands here, and that's a horrible alpha helix. So you have these 2 strands. This is A, over here is what? T. Here this is G, this is C, right? This is T, this is A, this is G, this is C, this is A, and this is T and T. So this is the sequence, right? So this strand has a different sequence than this strand, right? They're complementary. They're complementary, remember that. Even though this has a sequence, this has the complementary sequence. Opposite, if you will. All right. How do you make copies of this? So there's 3 ways. They thought, well, there's 3 possible ways, they thought. We could either have the old, let's call this one the old strand. Like I say, let's call this one strand and the red one's the other strand. To make copies of these 2 strands, there's 2 ways to do it. Or 3 ways to do it. It can be conservative. First of all, you have to understand these 2 strands have to open up, don't they? That's right. So there's 3 ways you can do it. We have the conservative way. Conservative means you keep the same, right? you conserve something, you keep it the same. So the conservative model would have you say, well, these are the two strands right here, they're both blue, and in the end, what's going to happen is these are going to open up, they're going to unravel, some machinery is going to come in and copy it, but then the two old ones are going to come back together again. So you're going to have this old strand, and then you're going to have a new strand that's going to be exactly the same copies, but both strands are new. Does that make sense? You still have A, G, T, G. The sequence is always going to be the same, no matter what method you use, but the two new strands are going to go together, and the two old strands go together. We call that the conservative model.
Isn't that what we say where like the old one is the parent strand, and then you have like the new one?
Yeah, one would be the parent, and one would be the new strand. The old strand, new strand is probably the best way to do it. But newly synthesized versus the original parent versus whatever, whatever, however you want to say it, right? But the bottom line is that you have the two old strands stick together, and you have the two new strands. We call that conservative. There's another way that you could do it. You could do it piecemeal, right? You can take A, G, and T, right? Let's make that blue. A, G, and T. A, G, and T, the first three here, and you combine it with a new strand, T, C, and A. And so you have one a part of an old strand, but then you have a new part, then the next part here is new. It's not, so you're starting to have parts of the old and parts of the new on each strand being copied. This is probably the most complicated, right? So you copy these in pieces. You don't copy them as a whole long strand, you copy them in pieces. So you'll take maybe you'll copy this piece, will get copied, and then the next piece will be new, and then the next piece will be old, and maybe the next piece here is new, is old, and the next piece is new. So you're copying them in pieces. So you get old and new mixed together. We call that dispersive. So you're dispersing the copying. You're copying in pieces, not as a whole strand. So those are the two ways so far, right? Either conservative or dispersive. So there's a third way, and that's semi-conservative. There's that word semi, that prefix semi again. You could take one old strand, and there's two strands, they're both red, and you copy it, but instead of having the two old ones stay together, two new ones stay together, or instead of having a mixture of old and new together in one strand, what you could do is you can keep one strand the same and then add to it a new strand. So you use this strand as a template for this strand. So then here, again, here's the old strand over here, and then there's a new strand. We call this semi-conservative, where you use the old strand as a template to make a new strand. Kind of like what we learned with RNA polymerase, right? Kind of like what we learned when we made an mRNA. We use the DNA as a template to make an mRNA. Well, the idea here is that we'd use the old DNA strand to make the new strand. But here we're copying DNA to DNA. Remember that. All right. All right, so that's not right. All right, so what did the scientists do to try to show which one of these? Because they knew that it was one of these, they just didn't know which one. Well, they had some tricks up their sleeves. They had isotopes. They had a test tube, and they had two forms of nitrogen. N14, which is lighter than N15. Does that make sense? And 14 is 7 and 7, and 15 is 7 and 8. So they have another trick. Has anybody here ever washed clothes? And you ever watch a spin cycle? You say yes, but I bet, has anyone here never washed clothes? Why am I not surprised? Hey, I was washing clothes. My mom had my brothers and I lined up to wash, fold, and dry and fold.
Based it for an hour, like you gotta put the black.
Exactly.
And then you're like, never mix color with white because then it can turn into.
That's right. That's it. That's it. My mother was of the opinion that she had children to help her do work. That's the whole point. All right, so, and I agree with that opinion personally. Why have kids if they're not going to help you? Yeah?
That's my philosophy. I help my dad build like decks for other people. Seriously.
Whatever your father wants, he's the boss. That's it. Our parents are our bosses. I'm 52 years old. My old man calls and says, do this. I say, okay. What are you going to do? They're your bosses. That's it.
My dad moved across the street, so they do that all the time.
Okay, so if you've washed clothes, you know that when it's in the spin cycle, what happens to it?
The water really fast.
It goes, the water drains out. Where do the clothes go? They get smashed to the side. It's called centrifugal force. Well, it happens that if you make it, you prepare the solution in a certain way, and you put it in a centrifuge. A centrifuge is just a spin cycle in a washing machine. That's all it is. Except you're not letting anything drain out. What you're doing is you're causing almost synthetic gravity. Have you ever seen the sci-fi movies where the space stations are turning? They're twirling round and round. The reason they twirl round and round is so that they can do what's called artificial gravity. And what you're doing is that you're spinning, so you're basically creating a spin cycle in the washing machine, but you as a human are sticking to the side, right? You're able to walk. Oh, that amusement park ride. All right, what is it? What is it? Yeah. What? It's, I think it's called the rotor or something. You like get in it, it starts spinning you around. You ever been in it? I mean, when I was a kid, I loved it. I got in it as an adult. I want to vomit every time, but I still do it because it's fun. So you're in it and it's spinning, and you're literally sticking to the walls. If you're small and athletic, I was never small, and I was also, even though I was athletic at one point in my life, I was too scared to do it. But people would stand up and walk on the side, I mean, it's crazy what you can do. You're walking on the wall, is what you're doing. So it's pretty cool. By the way, you can fall. So, and if the thing ever stops, bad things happen. But the bottom line is that force, it's called centrifugal force, can separate things by mass. So we can separate things that are more massive in a test tube if we spin them really quickly around and around. It's called the centrifuge. That's what the machine is. Usually they're small, but there are big ones for big sample liquids. And if you put two types of nitrogen on it, what you'll get is you get all the nitrogen 14 up here and all the nitrogen 15 down here. So you can actually separate by mass in a test tube. That's pretty useful in a laboratory. So if you have bacteria are only going to eat what you give them, right? If you have a plate of bacteria, and you put them on there, we call them plaques. By the way, that's why they call them plaques on your teeth, because those are bacteria. You have plaques, which are bacteria growing on here. These bacteria are eating the food that you provided to them on the petri dish, right? Does that make sense? So this is a gel. This is some kind of food source. You could use blood. You can use any kind of. It's called agar. The gel is called an agar. It's a seaweed. It's a seaweed gelatin. It's stiffer than Jello.
That's what they be eating on the ASMR videos. It's like
Yeah,
aloe vera.
Yeah, you can eat it, but I wouldn't eat it. It's seaweed. I like seaweed. I like dried seaweed, but seaweed, this agar, it's not tasty. You can add all kind. You can do it on this. There are some. Like I watch the cooking shows, and there's some famous chefs that do use agar as a thickening agent in food. The agar itself is not dangerous, but when we use it in laboratories, is as a medium to grow our bacteria. See, when you're growing the bacteria, this colony here is millions and billions of bacteria. So this bacteria is only using what you give them in the petri dish. So if you put nitrogen 14 in there, what is going to be in their nucleotides? Remember, nucleotides have what? Nitrogenous bases, which means they're using nitrogen. So what are the nucleotides going to be here? What are they going to have? Yeah. Nitrogen 14, because that's all they have to work with. So as they grow, more and more of the bacteria have nitrogen 14. You can do the same thing. You can do the same thing, except you do what? Do it again. In the experiment, they did it again. But instead of using nitrogen 14, what did they use? And they used nitrogen 15. So these bacteria here have nitrogen 14. These bacteria here have nitrogen 15. Are we clear on that? All right, so that's that. So if we centrifuge them down, the 15 would be heavier than the 14, so they would sink in. All right, so now that we understand that. So we start growing them in nitrogen 14, so they're light. I believe that's right. Did they start with 14 or did they start with 15? I can't remember. Grew bacteria cultures containing only nitrogen 15 for many generations, so that DNA was almost entirely composed of nitrogen 15. These chromosomes are dense, they're more dense, and spinning them down at higher rates means they're lower in the test tube, right? So we know they started off with nitrogen 15, they grew them in the test tubes, and when they separate the DNA or even if you just use the bacteria, they're denser, so they're going to be low in the test tube. Does that make sense to everybody?
Yep.
So they're going to be here, the nitrogen 15. All of the DNA was down at the nitrogen 15 level of the test tube. After growing the bacteria nitrogen 15 producing bacteria having chromosomes, they shifted the bacteria to growth conditions that were only on nitrogen 14. So they took that bacteria, they plucked a colony, they took a toothpick and they pulled the colonies off of this, and they moved them over an agar plate that had nitrogen 14 only, and let them grow there for a while. And what you got was not, so what are the possibilities? If they were conservative, what would happen? If each DNA molecule had the old strands had the old DNA, the old nitrogen, and the new strands had the new nitrogen, what would happen? It wouldn't have combined, so you would definitely get a band, right? If right, this hypothesis, if we're conservative, then you would get a nitrogen 14 band, right? And what else would you have from this new experiment? You'd have a nitrogen 15 band. You'd have the new DNA would have nitrogen 15. The old DNA would have nitrogen 14. We're good, right? Does that make sense? All right. Oh, jeez. They're just going to have to work with me, aren't we? If the DNA were dispersive, if it was the dispersive method, you would have what? Not a nitrogen 14, not a nitrogen 15, you'd have all the DNA would be what? Split up. They'd be right in the middle. They're somewhere in between nitrogen 14 and nitrogen 15. Well, what happened was, a really weird experience. In the first generation, you had halfway, exactly 100% of the DNA was in between 14 and 15. But in the second generation, you had some of the stuff, 50% was nitrogen 14, and 50% was in between. Half the DNA, all right, was found to be at nitrogen 14 levels, and half the DNA was in between 14 and 15. There was none at nitrogen 15. So that's an interesting result we'll have to talk about tomorrow, and then we'll get into we'll move on with the rest of the study guide. Please read that study guide and know what you're doing.
Are you gonna post this part, or you gonna wait till we finish it tomorrow?
I'll see where we get and how much time I have during the day. What would you rather I do?
Post half today and half tomorrow.
All right. So let me start again here for just a second. You have this bacteria that you're growing in nitrogen 15. So all the nitrogen that they have to make DNA is going to be found where if we spun it down? Here, right, in this area of the test tube. Do you agree? Because that's where we would find nitrogen 15. That part everybody understands, yes? Okay. Now, if we keep growing these bacteria, but now we transfer these bacteria over to this plate, which only has what? Nitrogen 14. They brought with them, the bacteria we transferred already had DNA, right? What DNA, what nitrogen did they use? Is that DNA made of? 15, right? As they transferred over here, now they're doing DNA replication, they're eating, they're doing binary fission, right? They're making more copies of themselves, they're growing, they're doing DNA replication. What nitrogen do they have to work with to make new DNA? Nitrogen 14. It's the only thing they have. So as they grow and multiply, all the new DNA, if it was a conservative model, if the model was conservative, in other words, you kept the old DNA strands together and you made two new ones, right? Then, what would you expect to happen if you spun them down? You took some samples from here, from this new plate, after you let it grow for a while, multiplying, and the colonies multiplied and grew, right? And you did DNA replication. Then you spun them down, you'd expect to see two bands. One at nitrogen 14 from those that DNA that was original, because it's conserved. Remember, conserved means you keep it the same, right? So the DNA here in this nitrogen 15, the original plate, nitrogen 15, you'd expect to see that there'd be some original DNA that's heavy. There'd be a band, right? And then the new bacteria that grew on this plate would now have another, the lighter DNA, right? So there would be a second band. That's what you would expect to see if it were conservative, if the DNA replication were the conservative model. Does everybody understand that now? Is that better? Is that better understood? Okay. What did this so now that's if it were conservative. But what would we expect to see if it was dispersive? We'd expect to see one band, right? We'd expect to see one band where? In the middle, right? In between the two. Because remember, what does dispersive mean? You take a little bit of this, a little bit of that, and you're taping together a new molecule, right? And so you get about 50-50, nitrogen 15, nitrogen 14, because it's all random. You'd end up having all the DNA pretty much have somewhere in between nitrogen 14, nitrogen 15, one single band. If on the other hand you had, what if on the other hand you had a semi-conservative model? What would you expect then? What if you had a semi-conservative model? Let's think about what that means. So, if you have a DNA molecule, and these are the original ones, they have what? Nitrogen 15, right? And then you're going to make copies now on nitrogen 14. You're going to have the original strand, right? But on that original strand, you're going to have this new nitrogen 14. So this half is going to be nitrogen 14, and then this half is going to be nitrogen 15, right? And you'd have two of those molecules. Right? So you'd expect to see, if we were just looking at the original bacteria with the original molecule, we'd expect to see one band at nitrogen 14, or 15, right? But now we have these new molecules with semi-conserved, so one old strand, one new strand. Where would we expect to see the band now? Would we expect to see, if we spun these down, remember they're sticking together, they're not separating. These two strains are not separating. This is one molecule, they're hydrogen bonded together, we're not separating them. We're just spinning them down. Where would we expect? Would we expect them at 15 or at 14? Yeah.
Do you think it might be in the middle?
They would be in between the two, that's right. We'd have one band in between the two, right? Just like the dispersive model. Man, that's harsh then, right? So how do we tell if we get a single band in between 14 and 15, how do we know it's dispersive and not semi-conservative? The second generation is the key. The first generation, you're going to have one band. You'd expect to see one band because of what I'm showing you right here. Half the DNA is 14, the other half is 15. You'd expect to see one band. But think about, and this is where this kind of multi-phasic thinking, right? Multi-step thinking. You kind of have to think outside the box. Think a little deeper than what you might be used to. Think about the next generation. Now you have, this is the first generation, right? Right here was the first generation. They made two copies. Now we're going to go ahead and do the second generation. So we had one cell here. Now we have two cells. How many cells are we going to have now? 4 cells. 2, 3, 4. So we're going to do semi-conservative replication, right? In the dispersive model, you'll always have a mixture, one band, always, no matter what generation, because you're constantly using whatever's there and you're piecing things together, right? Everybody agree with that?
You said dispersive.
And dispersive. In the conservative, you'll always have two bands. You'll always have one 15 and the other one 14, right? In the conservative model. That's the expectations. You expect that. If you see anything other than that, then you have to wonder what's going on. But in the semi-conservative, in the first generation of bacteria that grew on the new plate, right? After you move it over to nitrogen 14, you don't expect to see anything in nitrogen 14. You don't expect to see anything in nitrogen 15. You expect to see a band somewhere in the middle for the first generation. Now, what would we expect for the second generation? Let's take a look. The original would still be there because it's semi-conservative, so you still conserve one of the two strands, half the strands. So we go ahead and draw that. That's this one, right? Little one here. That's this thing here, right? And then you're making a new one on the nitrogen. The red represents the nitrogen 14, the new nitrogen source. So you have half and half, right? We're good? That would still be found where? Right here in this halfway mark, right? Okay. That's when this one's copied. But what happens when this one's copied, the nitrogen 14 strand? So you have the nitrogen 14 strand here, right? Do you see that? What's the new strand going to be made of? All nitrogen 14. So now you have a what?
One.
You have two bands now, don't you? You have one band, this one's going to make a band in the middle. Where's this one going to be found? On the top at nitrogen 14. So now let's go ahead and do the next one. That's what happens when you semi-conserve this DNA strand and this DNA strand. You'd expect to see two bands, right? What about this one? What's going to happen is you're going to get the same thing. You're semi-conservative, so you're going to conserve this strand, right? And you're going to conserve it. Do you agree? Yes? We're okay? And the new nitrogen, the new nitrogen 14 is going to have this, because that's the only nitrogen it has to make the new strand, right? But remember, what do you have over here? You have a strand that was nitrogen 14 in the first generation, right? But it's still there. It's not destroyed. What are you going to do? What's it going to make now? Another nitrogen 14 strand. So again, you're going to have two bands. One that's going to be nitrogen 14 only, and one that's going to be half nitrogen 14 and half nitrogen 15. So in between, right? So you expect to see two bands in the second generation and beyond, and from then on. And by the way, if you really think about it, you'd expect to see the nitrogen 14 band get brighter and bigger and bigger and bigger, because as you keep making more and more cells with nitrogen 14, the old DNA starts to become a smaller and smaller percentage of the DNA that's present. But anyways, the point is, this is nitrogen 14, this is nitrogen 15. In the second generation, you'd expect to see nothing at nitrogen 15, but you'd expect to see a band in the middle that's pretty consistent. This is going to stay pretty much the same. And then you'd expect to see a band of nitrogen 14 at the top. So you expect to see two bands. Is that making sense to everybody? This elegant experiment, it's elegant. It's a little more complicated than the mouse experiment, right? The mouse experiment was very logical. If this, then that, that's going to happen. This required a little more thinking, a couple more steps. But it's still beautifully elegant, isn't it? It's just really simple, straightforward. This is what I expect. If this is true, then I expect this. This did not happen. I did not get one band at 50% in every generation. So what do I know? Then what? Then it's not dispersive. I did not get with conservative, I did not get two bands, one at nitrogen 14, one at nitrogen 15. So what do I know? It's not conservative. In the first generation, I got 50-50. That didn't tell me I knew then, because I had one band in the first generation, I knew it wasn't conservative. But I did not know if it was dispersive or semi-conservative until the second generation. We finished that experiment and we saw two bands, and then what else? Then what did we see? Now what do we know? Because we have two bands. It's not dispersive and it's semi-conservative. Has to be. Otherwise you can't, how do you explain this? Does that make sense? All right. And that's the experiment. Those are the experiments that let us know that DNA replication is semi-conservative. The DNA is the molecule of inheritance. Okay. Classic experiments. One's Griffith, the other is Messelson-Stahl. I would expect as you might see these kinds of questions on an ACT or SAT. You certainly will discuss it and read it in the book. You'll read it and discuss it in college textbooks. You'll see it everywhere, okay? Because these are classic experiments. There are a number of experiments that people talk about in classrooms all around the world. Mendel. You've even talked about Mendel probably and his peas, the tall and the short, the yellow and the green, the wrinkled and smooth. Did you remember talking about that at all in middle school?
Yeah.
Well, some of you have. So some of you have started already some of these classic experiments. Mendel's experiments were classic. Darwin, you've heard of the name Darwin, yeah? Well, Darwin's an important name for evolution. We're going to get into evolution very soon. But Messelson, Stahl, and Griffith were very important people when it came to DNA. And not themselves, not the person themselves, but their ideas. All right. So the structure of DNA we talked about, and I think you've got it based on, the exam scores, based on your answers to. If you listen to this video when I post it this evening, you'll hear your answers to it. You're pretty solid on what DNA is, right? The nucleotides, ATGC, you know that the genes are on the nucleotide. So, and you read chapter 12, you know there's a test on it. You know that there's this thing called, instead of RNA polymerase, you have this thing called what? DNA polymerase. And there's a lot of steps in the book, aren't there? Did you notice I did not give you a lot of homework on what are the steps of DNA replication? What are the enzymes? I did not ask you about any of the enzymes or to write down a chart concerning the enzymes or the steps of DNA replication or which way is the DNA read and which way is it made. I didn't ask any of that. There's a reason for that. Even though I wanted you to read the chapter and be ready to test on it, I'm not going to ask it on my test, all those steps, because it's not on your state exams. At least it's not supposed to be. All right? And if we had extra time, I would go through it because it's very important, it's very interesting, and it's actually, very beautiful in what it does. I'm going to pause this now and I'm going to quickly show you a video on DNA replication just so you see it. But I'm going to say it again. Do you need to know the steps of DNA replication for the exam on Friday? No, it will not be on there. Right? I wanted you to read it. I still do want you to read it. I want you to take notes on it. I want you to understand that there are many enzymes involved in this. There are some qualities about DNA replication that I will expect you to know, and I'm going to go over them in a minute. But I want you to see DNA replication happen in an animation. And that's the video, that's DNA polymerase. And there's actually more than one DNA polymerase type. There's DNA polymerase 1, 2, and 3, and there's helicases and topoisomerases and just all kinds of three-dimensional thinking and maneuvering that you have to do to get the molecule to do what you need to do. And I think someone mentioned it yesterday that we have histones in DNA as well, so we have to unwrap the histones, and then we have to remake this, and wrap the DNA has to be wrapped up again. There's a lot of stuff that's going on here. This is not just a simple one step then another. But we don't have to worry about all that for this exam. You don't have to worry about that for your end of course exam. What you need to know is it's a semi-conservative process that you make exact copies. You do need to know the DNA polymerase, the enzymes, and the enzymes that are involved in making copies of the DNA have some interesting functions. The first is that they have correction. They have corrected spell. They have spell check. All right. They correct mistakes. That's important to understand. You can imagine if there's over 1 billion. If I told you to copy 1 billion words, a book with 1 billion letters, you can imagine. Even if there's just 4 letters, you're going to make a mistake, right? This process ends up making mistakes. What do we call those mistakes? Mutations. When we get a change in the DNA sequence, it's a mutation. So even though the process corrects mistakes, you still miss them. You always end up with mutations. Even though you have corrections, because eventually all the correcting in the world, you've had spell check for your whole life probably, right? When you're typing in a paper on a computer. I didn't have it when I was a kid. I had to use a dictionary and whiteout. So in any case, you've had spell check your whole life. Have you made mistakes with spell check?
Yeah.
Sure have. Even with spell check. Even though you have the computer telling you, no, this is wrong. You still made mistakes but of course less mistakes than you might have without it. So DNA polymerase has corrective features. So what does this do? This minimizes errors. All right. Now there's something about these we call them mistakes. And you make mistakes, and DNA polymerase makes mistakes. At the very molecular level, mistakes happen. So let me say this. Mistakes are not a bad thing. These mistakes, what they do, the mistakes, the changes in the sequence, the mutations, they increase genetic variation. The soul of life. Without it, we'd all be clones. We'd all still be single-celled organisms floating around in an ocean, a primordial ocean. We never would have had all the diversity that we have on the planet today. Yeah.
Would an example of a mutation be your eye color?
Eye color is a good example. Anything that you see there's a mutation. We started off, remember, as a single-celled creature. We were all prokaryotes. All life on Earth started off as prokaryotic. But because of mutations, because of changes in the DNA sequence over time, how do we know that? What evidence do we have that suggests that that's true? That's something that I'm going to talk about a little today, and we'll talk more about it as we go deeper and deeper into evolution, okay? When we talk about the DNA molecular genetic variation, we start off as a single cell, and very soon we ended up as a prokaryote. And then one prokaryote and another, there was competition, there was genetic variation. You ended up with a prokaryote with another prokaryote inside it, had some internal membranes. Then you started something that was called a eukaryote, a line that one of those prokaryotes ended up being eaten or eating another, and through a lot of small changes ended up being a eukaryote. Others stayed prokaryotes, and they changed into different kinds of prokaryotes, right? You have E. coli, you have pneumonia, you have staphylococcus, you have bacillus, you have all kinds of different prokaryotes, right? So you have all kinds of different mutations and changes that occurred. They have a big diversity of prokaryotes. And this bacteria swallowed another bacteria that has a really good ability to make ATP. It didn't digest it. It wanted to, but it didn't. That's probably why it took it into itself to digest it, but it didn't for whatever reason. And what you end up with is something with a membrane surrounded by another membrane. So remember how do cells take in other big things? How do cells take in big things? Phagocytosis, that's right. So if they take in this cell, if this big pre-eukaryotic cell takes in another cell like this, what's it going to have? When it's inside, so here's the cell, now it's inside. What's it going to have? It's going to have a piece of this membrane, the black membrane around it, right? And it's going to have a blue membrane inside. It has its own membrane. So it has a double membrane. It's really good at making ATP. What do we call that? A mitochondria. That was the first eukaryote. This is called and this is something that will be on the test. Endosymbiosis. Symbiosis means a very close relationship. Endo means inside. So a close relationship inside. How do we know this? How do we know that this happened? Well, first of all, you need to know that there's two membranes to mitochondria, so that's really interesting. Why would anything have two membranes? And then secondly, they have their own DNA. Did you know that? And they have their own ribosomes. They have their own DNA. Mitochondria have their own DNA. It's different from our DNA, from the eukaryote DNA. And the ribosomes are very, very prokaryotic-like. They're different sizes than the ribosomes that are in the cell itself. So it's two separate DNAs, two separate sets of ribosomes. That's interesting. And when we look at the genes in the mitochondria, they're very similar to genes that we find in prokaryotes. So we can find a lot of similarity between mitochondria and bacteria. And in order to explain this, we say, well, it probably was some old, ancient bacteria swallowed another bacteria that could do it, did phagocytosis, that created that double membrane. And then it had its own DNA, was not digested, but what happened was that the big bacteria was good at getting food, at getting sugar, and the little bacteria was really good at making ATP. And they got together and did a partnership. Of course, every cell after that had a what? What did every cell after this have? Mitochondria. Every time it did binary fission, the mitochondria does its own DNA, its own replication, and you have many different kinds, and then you have the cells making their own. Oh, wait, what does that? The cell's doing its own DNA, its own thing, and you have always from then on. These cells have mitochondria. So that's why we find every eukaryotic cell on the planet has a what? Mitochondria. Every one. Now time went on, probably thousands if not millions of years, right? Probably maybe thousands of years. These aren't animal cells yet, by the way. They're probably not even eukaryotes yet. They just have mitochondria. But these are the progeny of eukaryotes, right? So these are going around, and now they become eukaryotes. They have a nucleus, they have organelles. Who knows what kinds of organelles? We don't know. We can't tell, right? We can just all we can do is look at the evidence we have in front of us and compare what we see and make educated guesses at what's going on. Nobody has a time machine, right? So one of these little bacteria could do something. There was these bacteria that we know existed, because they still exist today, by the way. We call these things cyanobacteria. They do, do you know what cyanobacteria do? They do photosynthesis. These bacteria change the planet's atmosphere. Before the bacteria started multiplying and doing what they do, before that happened, this Earth's atmosphere did not have much free oxygen at all. How can we tell? How do we know that the primordial Earth had very little? Why do I say that? Am I just making that up? Am I telling you a story? No, there's evidence. We can look at the rocks, and what happens when oxygen comes into contact with iron? Rust. We can look at the rocks that are in the earth itself. The evidence is literally in front of us. And if we go back far enough, we go dig deep enough, what we find is that the rocks are not rusted. But then, as time went on, all of a sudden, as we go further and further, still deep in the earth, further up the layers, all of a sudden the iron becomes what? Rusted. How do we explain that? More oxygen is available. What's the only process we know that could free the oxygen? Photosynthesis. So this photosynthesis was done and by the way, we also have all kinds of other evidence. We have fossil evidence of these cyanobacteria. They created these little mounds. I could show you, but we don't have time for it, unfortunately. They created these amazing mounds all over the planet. We see them in the ocean, where the ocean surface, bottom of the ocean has come up to the surface in places where there's been uplift. We can see the fossils of these cyanobacteria, so we know they were there. And this is billions of years ago. This is before that we have any fossils of any other creature. No multicellular organisms, just prokaryotes. So these little bacteria changed the planet's atmosphere. This planet used to be purple. You know, we call it a blue dot. because the sky's blue, right? On a non-cloudy day, right? Right? You've seen the pictures of the Earth from space. What color is it? Mostly blue, maybe some white, right? It used to be purple. So the planet surface, the color of the planet, the atmosphere itself, all changed because of these cyanobacteria. So there's these photosynthesizing bacteria. Here's this cell that's really good at capturing sugar and eating things, right? Maybe it has a nucleus, maybe it doesn't, I don't know. But here it has a mitochondria, right? Here's this little bacteria that can do photosynthesis really well. What do you think happened? And it happened again. Oh, look, yummy, yummy. I'm going to eat this green thing. I'm going to eat my vegetables. And when it does it, of course, what's it going to create? It's going to create a double membrane, right? Here's a double membrane here from this bacteria. Originally, the mitochondria. And of course, it has its DNA, has its other organelles, who knows what they are. But then now you have another double membrane, don't you? And guess what? The chloroplasts also have their own DNA. The chloroplasts also have their own ribosomes. The chloroplasts are very similar to bacteria as well, in many different ways that we're not going to get into. So this again is another example of endosymbiosis. And this also explains, it's a beautiful theory, because it also explains why every plant has both mitochondria and chloroplasts. Because all eukaryotes first had mitochondria, and then some of them developed chloroplasts. Of course, this was the original plant cell, this was the original animal cell. Some of these went on and became animals, all the animals, including us. And these went on and became every plant, right? Trees, grass, what have you. All this because all this happened, and all the diversity you see in a forest, and the jungles, and the earth, the oceans, all of it because of what? Mistakes. Mutations. There was also a lot of death. You have to understand that. When mistakes happen in DNA replication, usually what happens? What happens if there's a mistake? Well, usually actually nothing happens. There's no right, silent mutations, because remember the genetic code is redundant, yes? There's more than one way to get the same amino acid. We learned that. You have these things called silent mutations. If the mutation doesn't happen in a gene, it might not have any impact at all, right? So there's a lot of redundancy in the genetic code. So that's one line of defense. So even though we get changes, but you can think of that as prepping the DNA for a change that might occur that might be successful. But even those, if you get a genetic mutation that happens in an important gene, what's likely going to happen is a disease. Likely you'll get something like cystic fibrosis, sickle cell anemia. And I mentioned sickle cell anemia for a reason. It turns out, and I've taught you this already, is sickle cell anemia all bad? No, it's not. What advantage does it give you?
White blood cells.
First of all, it impacts red blood cells, hemoglobin, right? Not white ones. But secondly, think about it. Think about what I had taught you. You don't know this, but so there's a major killer that's been killing people, cows, mammals, for millions of years. It's a killer. It's a tiny little worm, little parasite, called malaria. All around the world, if there's mosquitoes, malaria has a life cycle where it lives in a mosquito and it gets transferred by a mosquito sting or bite. So malaria goes from a larvae into the mosquito. It's actually a specific kind of mosquito, by the way. And from the mosquito, then it goes into the animal. And I say animal because this does not just kill humans, it also impacts cows and goats and what have you. Anything with red blood cells, right? Because it goes into the red blood cells, animal, and then of course eggs are laid and it goes back to the streams and back to the mosquito. The cycle continues. This is a killer. It kills millions of people. It's killed billions probably in the history of humankind. It's wrecked civilizations. It causes strife, hunger, because it kills the cows, the food sources for the people as well. Horrible disease. It's forced evolution. It's forced humans that in order to survive. If you got malaria, you're not guaranteed death, but it's possible, and you're certainly going to get sick. So what happened was that this mutation occurred. You have sickle cell. If you have the disease, remember, let's do a Punnett square. Can we do a Punnett square? Have you done Punnett squares before? I think a lot of you have, but let's go ahead and do it. Let's call sickle cell, big S is normal, so you have normal hemoglobin, and little s, let's call it normal. When I use the word normal, I mean average. I mean non-diseased. Are we clear? I don't want to make it sound like, oh, these guys are abnormal and you're normal. That's not what I'm saying. Normal just means not diseased. You'll see it used in science a lot. You got to get used to that. Right? So normal. Little s is sickle. The cells are sickled. It's a sickle cell trait, right? The cells aren't necessarily sickled, but it's a trait. Well, it turns out, if we do a Punnett square, and we do two heterozygotes, this person has the trait, not the disease. The cells are not sickled. They don't get stuck in the capillaries. There's no strokes or events that occur to these people that have the trait. So if you know someone with the trait, they're likely have very little or any symptoms at all. If they marry someone with the trait, is that clear which is uppercase and which is lowercase? Okay. 1/4 of these people are normal. 1/4 half are going to have the trait, and 1/4 are going to have the disease. Unfortunately, historically, the people that have the disease die. And you might think, well, why? How is it that we still have the gene, right? Because we should have all the homozygous recessives, right? That's what they call these homozygous recessives. They're all dead. So why is this not disappearing from the population, right? Because you would think, well, they're dying, so every time you see little s, those people die, they can't have babies, so how do we get the little s into the next generation? Yeah.
Is it because that those two still carry the trait of the little s?
That's right, the little s is still here, you see it? It's in the heterozygotes. But you know what? What's interesting is what happens to the big S, big S. Again, back in the day, today we have some treatment. We have some sulfa drugs that help, but unfortunately still no cure for malaria. This little worm, we can give you drugs that will help stop the infection right now. It gets in your liver and stays dormant in your liver cells. So even though you feel better today because I gave you drugs, in a couple months, in a year, 2 years, in 5 years, you may get stressed out and that little bug wakes up and starts killing your liver again. And you get sick again, right? And die or get sick and you need more drugs. So it's a really long-term issue. Well, what turns out that a lot of people that don't have the trait, what happens to them? Back in the day, talking about before modern medicine. What happened to them? Yeah.
They would probably get malaria.
They get malaria and die. And likely that they would not have babies either because they would die before it's too late. So what happens to the population? What do we see? Where there's a lot of malaria, what do we see? A lot of sickle cell genes, about a lot in the population. Why do we see a lot of sickle cell genes in the population? Because it's an advantage. Because you get to live. If you don't have it, you don't live. And people like to say, well, sickle cell is a high, you find a lot of it in African American population because people are from Africa genetically, and that's where you find a lot of malaria in Africa. That's true. And it is true that sickle cell is an issue for the African American population, but what you also have to understand is that anyone from the Mediterranean has the same issues. Anywhere there's malaria, you're going to have the same issues. And guess what happens anywhere there's malaria with humans? What do you find? Sickle cell, or a variant of sickle cell. There's another variant of sickle cell. There's more. We talked about, I try to teach you that the red blood cell has how many different hemoglobin chains? 4. There's 4. 2 alphas, 2 betas. So if you get the same changes, there's 4 proteins, 4 genes, where you can get a mutation in any one of those 4, and you get the same result. And what the advantage is, this malaria, if you have the trait, malaria cannot attack your red blood cells. Oops, you just block them. So it's an advantage. So anyone from like Greece or Italy or the Middle East even, that's by any large group of any water population, it includes Jewish people and Arabs, and you have the possibility of having this higher propensity of this gene that allows you to survive this horrible disease. Malaria is it's even in the Old Testament Bible. It's a plague, right? So this trait helps you survive it. So this is an example of a mutation, a mistake, that's an advantage. Mistakes also have led to, someone said, eye color changes. That's right. Otherwise, we'd all have the same eye color. We'd all look like each other. Why do we look different? Because of the genetic variation. You have all these possibilities that build up in a population. And once you have genetic diversity, then natural selection can take hold, and you can get populations changing over time. Yeah.
Doesn't it also cause variations like disease and stuff?
Say that again.
Kind of like also, just like it causes like eye color, and like does it also like cause disease?
The changes, the mistakes, can they cause disease? Absolutely. And we have an example right here. It causes disease. These people are dead because of it. These people are dead because they don't have it. This is just a really great example of both that a mutation is by itself. Somebody says you have a mutation. That does not mean it's good or bad. You have to ask yourself, well, what does that mean? It's mutated, so what happens? So now what? What's the result of the mutation? Does having blue eyes, is that a problem? It is if you're in Africa or Australia or even South America. The sun is very bright. Is being light-skinned a problem? It is if you're in Africa, South America, or Australia. And in fact, Australians have some of the highest percentages of skin cancer in the world. Because you understand, the modern Australian, not the Aborigine, right? Not the Aborigine. Aborigine is dark. An Aborigine person is generally darker skinned. The people in Australia, do you know that they were colonists? They came from Europe, from England and the United Kingdom. They were prisoners that were sent there. It was a penal colony. Did you know that? So those Australians did not evolve there. Don't put anything away yet. Those Australians did not evolve there. So they're exposed to all this bright sunlight, but their skin is still light. So they're not adapted. Is dark skin a problem? It is if you're up north. If you're in Cleveland, you can't absorb vitamin D. You can't absorb sunlight. You need to have sunlight actually penetrate your skin and interact with your blood in order for you to make vitamin D in your blood. So what we find is a lot of people here in this latitude, their darker skin, end up not being able to, not having enough vitamin D in their blood, which, by the way, can lead to increased risk of diabetes. Isn't it interesting that diabetes is a problem? So vitamin D and all kinds of impacts happen, and so the differences between us can be positive or negative depending on the environment. That's the lesson. In itself, being dark-skinned or light-skinned, blue-eyed or brown-eyed, whatever, tall or short, fat or skinny, by itself is not good or bad. In a particular environment, it could be an advantage or a disadvantage. It all depends. All right, we'll finish this tomorrow. Have a good one. This gene, this chromosome, this DNA molecule is long. In the humans and in mammals, they're linear, and in bacteria, they're circular. But in any case, there's this long message, but the entire molecule is not a message, right? There is a section of the molecule that we call genes. So in this particular example here, only the green sections would actually have a gene. And each gene would be giving a label, LAC 1 or BAC 2 or whatever, right? It would represent what protein is actually expressed. So when this gene leads through transcription translation to a protein, when that DNA sequence leads to a protein, we call it a gene. Is that clear? Is it?
Yeah.
Okay, so we call that a gene. And everything else in between, for a time people thought it was junk. It's just leftover from evolution. When today we understand it's much more complicated. Today we understand that those areas of the DNA that don't code for a gene are very important in regulating these genes, in turning these things on or off. So that when we're talking about genes and we're talking about inheritance and DNA, we also have to talk about the idea that as humans, and not every organism is this way, but as humans, we are what are called diploid. Diploid means we have 2 copies of the DNA, 2 copies of each chromosome. 1 copy comes from dad, 1 copy from mom, right? Male and female. Together, they combine their DNA. And actually, we should say recombine their DNA, right? Does that make sense? Do you understand the word recombine?
Mm-hmm.
What's the difference between when I say combined and recombine? What do I mean by that?
They were combined once before.
Yeah, when were they combined? When mom and dad were made, right? So every time, there's this word that kids use a lot and they don't really understand, but there's this word that we all use, and it's called sex. And in biology, that's what sex means. It's mixing the DNA. Sex is for procreation, biologically speaking. There are other benefits as well, but the bottom line is that when we're talking about sexual creatures, things that not everything has sex. Is that correct? Do you understand that? All right, some things multiply through binary fission, for instance. But things that don't have sex, what they are is they're clones of one another. When you get a bacteria doing binary fission, they're cloning themselves. There is no there's very little genetic variation. So if you have a colony of bacteria, they're all pretty much the same. When there is a difference, now interestingly, bacteria still find a need to increase their genetic variation, so they have a different way of doing it, but it's not really sex the way we understand it. So sex, in the biological concept of sex, is just simply taking and recombining your DNA. Randomly choosing one of these. Now you have this parent here has 2 copies of all. How many chromosomes do humans have? That's right, 46 or 23 pairs. That's for humans. You need to know that every organism has organisms have different numbers of chromosomes. An ape, I forget how many apes have, or bananas, or wheat, or whatever. Each organism has a different number of chromosomes. And it's very specific to that species. Humans have 23 pairs. So this is, you're going to choose one copy. Either you're going to send the black or the blue copy of your chromosome to your child. You only get to send one. That's why we make these things in the male, we call them sperm. In the female, we call them eggs. They're called haploids. The word loid refers to, or the suffix loid, the base root, the root word loid, we're looking at chromosome. Is that clear? So diploid means what? Diploid. So ploid means chromosome. Ploid means chromosome. So diploid means two chromosomes. And haploid means what? Half. Half the chromosome. So instead of two, these kinds of haploid cells, the only cells in your body that don't have 23 pairs, by the way, only have half the DNA. They have half the number of chromosomes. Is that clear? I hope you wrote that down. I hope you understand that. These are called, these other cells, the rest, how many chromosomes are there? How many other cells? If these are haploid cells, what are the rest of your cells called? Well, diploid, that's right. These are called, another word for these haploid cells are gametes. And yes, you have to know all these words. Gametes. All right, so a gamete is a cell that has half the DNA that the rest of your cells have. Another word, and yes, you have to know all three, is sex cells. Right. Cells are used for sex, for exchanging DNA. So they're called gametes, sex cells, or haploid cells. The rest of your cells are in your body. They're in your body. So what do you think we call them? Besides diploid, what else do we call them? Say what? What do we call the diploid cells? The rest of your cells in your whole body. There's 1 trillion cells in your body. Maybe a female has 10,000 eggs. She's born with them. You were born, ladies, you were born with, if everything went right, you got about 10,000 eggs. That's all you get the rest of your life. Men make millions and millions of sperm all the time. We're constantly making sperm till the day we die, till we die. You ladies have plenty of eggs, right? 10,000 eggs. You can lay, not lay. you can release one egg a month, and that's what you do. By the way, everybody understands that your menstrual cycle is attuned to the release of one of these haploid cells, right? It's this haploid cell that when it releases from your ovary every month, it releases hormones that tells your body, thicken the uterine wall. Get ready, I might be coming. That's what it's doing. It's sending hormones saying, start to send blood vessels, get ready to maybe make a placenta, we might be getting pregnant, get ready. So your menstrual cycle is literally preparing for your child to be born. If that cell is not, if that egg, that gamete, that haploid cell, is not fertilized, if it does not meet a sperm, it does not become diploid. By the way, sex is about the haploid cell becoming diploid. Does that make sense? When it becomes diploid, it's a full creature. Until then, it's just an egg. It's half the cell. Half a human being, literally. Okay, half the DNA anyways. If that egg meets a sperm, then the process continues. It starts to do mitosis, it starts to embed itself, it digs itself using enzymes, it digs itself into you did this when you were being conceived and developing. I did this. We bury ourselves into our mothers. We eat our way into the mother's uterine, and we start to send hormones that start to connect the mother to the embryo through what's called the placenta. We start to develop specific tissues, and there is the womb. And the womb is not in your stomach, the way some little kids will say, oh, it's baby's in the stomach. It's not in the stomach. If you were in the stomach, you'd be eaten. The stomach is for eating. You're in a womb, in the uterus, right above the vaginal canal on the other side of the cervix. All right? So that's the basic anatomy of what we're talking about here. So this haploid cell becoming a diploid cell is the key to that further development and then finally birth 9 months later. Now, if that egg is not fertilized, it dies in about a week after it's released. Give or take a few days, whatever. If it's not fertilized, the egg dies. That means it stops producing hormones. That means all that blood, all those blood vessels and tissues that you started to develop, your uterine wall was getting thicker, getting ready for implantation, right? The hormones aren't there anymore. So what happens? All that tissue gets released. And so that's what you call a menstrual cycle. That's the release of that's why you bleed. That's the thickening of the wall that's no longer needed. Your body says, I don't need any more, we're not pregnant, it's cool. And out it goes. You cramp up because your muscles are trying to squeeze out that tissue because it's all there. You're trying to get rid of it. You don't want it to sit there because what happens to dead tissue? It can, but what happens to dead things? It rots. It starts to release enzymes. Lysosomes start to break. And that's no good for your body. Does that make sense to everybody? Okay. So it's a good thing and that's also why women, when they go through menopause, start to feel like, they call it a change of life. There's a whole emotional and psychosocial event that occurs when that happens, and you should be respectful if your parents go through, your grandparents. When they're going through that, they're having a change of life, right? They're becoming, in essence, they're becoming less fertile, right? And in our society, that starts to impact your psyche, doesn't it? Yes? Mm-hmm. Right? It doesn't?
Yes, it does.
You would know? Really? You would not know either? That's interesting. If someone said that you would have to be castrated, why would you not understand then the impact of that? How would you feel about that? Yeah, that means the removal of testicles. Removal of testicles.
It doesn't actually remove the testicle. Doesn't it just like remove the tube?
It removes both of them. If you remove both testicles, your castration, what's the problem? Why do you care?
I wouldn't.
You would.
Besides the pain, but like after that, I wouldn't care.
Yeah, you would. You say you did, I think you're saying you wouldn't because you're being obstinate. Yeah, you would.
Why? What's the point of if you want kids? But like.
Okay.
That's not in my future.
Okay, so you're not. Let's just say this. I would tell you to be reflective, right? Be reflective. Think about what it is to be who you think you are, what it is to be who you are, and understand that if you lose your testicles, you will have a definite change in who you are. You will no longer, the day after a few days after, probably the day after, and certainly a few days after you lose them, your whole body will change. Your hormones won't be rushing through you the way they would. The things you want, you won't want anymore. The things you thought you liked, you won't like. Your tastes will change. Your perception of the world will change. So, although menopause is not castration, okay, so let's be clear of that. A hysterectomy would be more like, a complete hysterectomy would be more like castration for a woman. Do you know what a hysterectomy is?
No.
It's a removal of the ovaries and the uterus, if it's a full hysterectomy. So that's done often because of cancer, right? Cervical cancer, uterine cancer, ovarian cancer sometimes requires a removal of ovaries and uterus from a female. That can, it would be more like castration is for a male. Yeah.
What about like, what about like a vasectomy? What would that be?
Vasectomy is just tying the tubes. All you're doing is stopping the sperm from getting out of the body. So that there's a difference between, you're still producing sperm. And psychosocially, what that means is you still are capable of making babies, and on some level that feels, for a lot of people, feels better than never, making that final choice of never being able to produce babies again. But understand that it's not just about producing babies, right? Like you say, you don't want to make babies and you'll never see that, and that's fine. But it's about changing because those hormones do a lot more than produce babies. Testosterone does a lot more, estrogen does a lot more than just getting ready to produce babies. It's a very broad signal for your whole body. So that changes who you are. And so people that go through these life changes, it really does impact them. Often, not always. Some people, my mother-in-law went through it and was like, whatever, moving on. But some people, although she has gone through depression, so maybe depression that's part of it. I don't know. So you got to be careful, and if you have people in your family that are going through this, you should be considerate. That's all I'm saying. Yeah.
How do they like transgender people? How do they give estrogen or testosterone if it's like a?
It's a very good question. You give them through injections. You take injections.
Where do you get it from? Like other people?
I see. Estrogen is a protein that we have the genes, so you can make, I'm not sure exactly how they make it. I can tell you they used to remove it from things that, like, for instance, insulin used to be removed from pigs, which led up to a problem for Jewish people and Muslim people if they were diabetic. Like, how do you take pig insulin, right? Either you die or you take pig insulin. Yeah, and some people would choose that. Others would choose other options. The bottom line is, what are you going to do? You say you would die. Would you let your baby die?
That's a tough question.
Yeah, that is. You're a father. You're a Jewish father or a Muslim father or a Christian who doesn't eat pig father, and you don't believe, you think pigs are unclean, and the only way you're going to save your baby is to give them pig insulin. Now you got not see you. It's easier to make a decision for yourself. It's much harder to look at your baby, your brand new boy or girl, and look him in the eye and say, "Well, you got to die." Right? So there's a whole. Things get. All I'm trying to say to you all about all these decisions that we. And I remember being in your shoes. It was very black and white. When I was your age, life was very black and white. What I'm saying to you, in other words, things were clear to me. This is right. That's wrong. You're stupid. Why do you even think that? I don't understand you. Dummy. Right? But what I'm trying to say to you is that life is a lot more complicated than you give it credit for. And you need to just be reflective and think. And maybe you're right. Maybe it is clear. And it's good that you have an opinion. Now, I'm not saying your opinion is not valid. I'm saying think about it and think about all the options. Not just the ones that you see right in front of you today. Like you say, you'll never have a child. How do you know? Right? You may choose when you're 20 or 30 to have a child. That may be a choice you make. All right? You don't know how you're going to feel in 5 years, let alone 10 or 15 years. And that goes back to the original discussion. When you make final decisions like castration or hysterectomies, now you're making decisions that you'll never be able to reverse. You can't go back after it's taken out of you. You've lost that piece of you. So even if you never do have children, there's the possibility of children. And so that is something that some people cherish. Others don't, right? So it's like, I don't have children. I will not have children. It's not an issue for me. But there is the possibility, even though I'm 52 years old, to have a child now would be inappropriate, right? I'd be 70 when the child's 20. That's not right. To me. To my father-in-law, that was fine. He chose to have my wife, and I'm glad he did. So the point is that everybody has their own choice, and you don't know where you're going to be in 5 or 10 years. So think and just be reflective. All right. So anyways, haploid cells, diploid cells, the parent is going to send one of the 2 copies. Where did they get these 2 copies? From their mother and their father. Where did they get those 2 copies? From their mother and their father. For 23 chromosomes. Is that clear? 23 chromosomes. So, and every species is different. So for us, it's 23 pairs. For other organisms, it might be 100. Who knows? There might be 5. It might be 1. Whatever. Okay, so when we're talking about 23 pairs, we usually, what you'll see me note, what you'll see me write over and over again for the next week or so, is you'll see me write the number 1 here and the number 1 here. So every chromosome has a number, numbered 1 through 23. Is that clear? So this is number 1, and every chromosome has a different shape. In fact, I have a poster up on the walls somewhere. That one's falling down, but somewhere on the walls, I have a poster. Maybe I didn't put it up, but that has all the different chromosomes and the diseases that happen when you get an odd number. All the numbers here are even. Do you see that? They're all number 2, specifically 2. There's 2 of each. So 1, 2. There's 2 number 2s. I shouldn't put 2, 2. I should put 2 number 1s, 2 number 2s, 2 number 3s, etc., all the way up until 22. All right, I'm sorry. That's not true. 21, yeah, I'm right, 22. There's 2 of each. The 23rd pair is odd. It's a strange pair. Strange pair. Now let me say this, for numbers 1 through 22, we call these things autosomes. They are body chromosomes. There's chromosomes that are for running everything that goes on in your body. Maybe this here is this, let me redo this. Let me reduce this. Maybe this here is the region where is the gene for eye color, or one of the genes for eye color, because it's actually, eye color is controlled by more than one gene, so that's interesting. Maybe this is the gene here that controls insulin. Maybe this is the gene that produces channel proteins, glucose channel proteins. Maybe this is the gene that regulates estrogen in your body. And by the way, males have estrogen as well. We produce estrogen as well, so you all know that. And females produce testosterone. So it's not as simple as people like to say, you have too much testosterone, you have too little estrogen. Here, maybe this is the one that produces, gabapentin, GABA. Anyways. So anyway, each one of these is producing a different protein. Is that clear? But there's 2 copies. So let's look at what we've talked about before, right? What we've already discussed, sickle cell. So one of these is going to look at maybe this one here on chromosome number 1 produces this enzyme right here, this region of the chromosome, this gene produces hemoglobin. This one produces normal hemoglobin. So this is the big S. Remember the big S, little s from Punnett squares? This is what they mean. This is what it actually means. Big S is the gene, the version of the gene, the genotype, the type of gene, right? Genotype, the type of gene that's dominant. If you see it, if you have this version, you're going to see it. This is going to be the trait that you see. The trait is what you see. The genotype is the type of gene, the sequence. And where this version over here, let's do it in a kind of a green color. Let's say this over here is little s. If you have 2 copies of this version, if you got one of these copies from mom and another copy of this version from dad, then you have sickle cell disease. If you get one of each, then you have your heterozygote, right? And you have the trait, but not the disease. And if you have 2 big copies, you don't have the trait or the disease. And that is, when we start to talk about Punnett squares, you have to keep that in mind. The chromosomes have on them gene sequences that produce proteins. Some of them will overshadow others, even though they'll both produce proteins, right? Both of these are going to produce a protein. This one is what you see more of because it works. This one doesn't work, but it doesn't show up. There's no symptom. There's no trait that you see with your eyes or consequence from having a small one. Yeah.
So Down syndrome is just that 23rd thing happening.
Yeah. It's a little bit. I'll get to that. Down syndrome is when you have an extra chromosome, number 21, not 23. So Down syndrome is when you have one extra chromosome. See, the thing is that this is all balanced. Remember, what are we talking about? You gotta just keep going back to your basics, right? You have to think of the process. It's all about process. If you understand the process, you understand the end result. You can apply that process. The process we're talking about here is producing a protein. That protein does the function, right? All right, so let's say that let's make an analogy, see if it helps. Let's say that we make windows, right? We have a process that makes windows, and we're building a room. And we end up, instead of building walls, we make all our whole room, our whole building's made of windows. Will that work? No, it'll collapse, right? So it's the same in your body. You have a certain number of, these things are producing proteins. You need to be making 2 versions of it. There's 2 genes, and your body's adapted so that you're producing, the amount of proteins that are produced by both those genes is enough. What happens if you produce too much? It's going to be unbalanced. You'll get mental retardation, growth stunting, overgrowth, enlarged heart, enlarged lungs, brittle bones, who knows? It depends, all kinds of diseases, depending on what? What are the genes on that chromosome? If I have more than one, if I have 3 chromosome number 21s in case of Down syndrome, then it leads to having mental challenges, developmental challenges. If you have more than one chromosome number, if you have 3 chromosome number 1s, you're probably not even born. So many things go wrong that you miscarry. In fact, there's very few of these chromosomes, these 23 chromosomes, that you can have more than one of, or more than two of, that allows you to survive. I'm going to talk a little bit more about this once I introduce you to the 23rd pair. So they're very interesting. Yeah.
Okay, so if you have like 3 of the 21, is that like a mutation?
That's a fantastic question. It's not a mutation, right? So you're copying, mutations occur when the DNA, something goes wrong with copying the DNA, right? Instead of an A, you put a T, instead of a G, you put a C, you put an extra piece of it. That's a mutation. What happens with having an extra chromosome has to do with mitosis, so we have to talk about mitosis first. It has to do about separating the chromosomes. When you make those haploid cells, something goes wrong. Instead of taking those 46 and putting them making two 23s, you make one that has 24 and one that has 22. Now you got problems. The 22 probably won't survive. The 23, if you have the third chromosome the extra chromosome is number 21, you have Down syndrome. Yeah.
Then is something similar like this, is that how autism occurs?
Autism is much more complicated. So there's a developmental issues. My understanding of the research, which is from a layman's point of view, I'm not a physician, but my understanding is that autism starts in the womb. It's developmental. There is some linkage to genetics, but not much. Older fathers tend to have, we do see an increase in percentage of autistic kids from older parents, older fathers specifically, not older mothers. So the details of autism are still, and also autism is a spectrum. So it's kind of hard to, like, there are people that are normal functioning, like me, regular, you wouldn't even know, and they're slightly autistic. And they could be in the spectrum. It could mean that they're geniuses, because you can have what they're called savants, right? There's a really good show that my wife and I like to watch. It's called The Good Doctor. I don't know if you've seen it. Yeah, so I love the show. My wife and I are catching up, so we'll talk about it. And it's about a doctor who's autistic. Now, is it realistic? Probably not. But could there be someone who's on the spectrum that's a physician? Sure. That particular show that has the doctor, on the spectrum, but I just can't see someone who's a surgeon flipping out in the middle of surgery and they be like allowing them to become a surgeon. I'm not sure that would be okay with people today. So I'm not sure. So, but it is possible to have someone on the spectrum. For instance, there are people with Down syndrome that are more intelligent than a lot of people think. And they're actors. You'll see them acting, and they have more money than I do. As far as that goes. So when it comes to these things, it's always difficult to pigeonhole, to put a finger on it. Again, back to what we said earlier, it's not black and white. There's a lot of gray area. There's a lot of gradient. You just kind of have to take people for what they are and talk to them, and let's see if it works or not. Yeah.
If Down syndrome is an extra 21 chromosome or whatever, is there a way that if they figure it out early in pregnancy, could they do something to prevent it?
No, because that's in every cell of the body. And we can't, up to this point, maybe in your generation you'll figure out a way to do it, but up to this point, we can't reach into a cell and pull one chromosome out. We can turn one off, maybe. That's something you might be able to do. But we have to understand genetics a little better than we do today to be able to do that. And we'll talk about turning genes on and off soon, probably next week, okay? Yeah.
So they did a test on every single chromosome to see which one made Down syndrome.
Man, you guys are on the ball today, asking some really fantastic questions, making some really great observations. Yeah, we know where a lot of the genes are, and we're still learning. We're still trying to pinpoint where all the genes are. We've sequenced the whole DNA, right? So we know what the code is on every single chromosome, but we're not sure where all the genes are. We have some estimates of there being about 10,000 genes or something like that. But we're not sure exactly what each one does and how it impacts. And it's not just one thing. Remember, I keep trying to tell you that we are a system, right? It's not just, gosh, I have blue eyes, so I have the recessive trait. We're done, right? So that's one gene. You're more than 10,000 pieces. There are probably a few million different pieces and parts to you that have to work together, right? So how do 10,000 genes control millions of character traits? That's a whole nother issue, okay? Yeah, anybody, I don't care, go ahead.
If like say like a dad had like the sickle cell trait, but he didn't actually have the disease, and the mom doesn't have anything, but the son ended up getting it, how does that did the mom activate the trait or something?
That's a long story. We'll talk about it a little bit better after we get done with this, okay? It has to do with more, try to write it down and remember to ask that question again. Yeah?
What sort of genes lead to people having some forms of albinism, including having like purple or violet eyes?
Albinism is purple eyes. I'd have to look that up. I'm not sure. Red eyes. Albinism is the lack of pigment in your body. Everyone in here has pigment in their body. The whitest among us has pigment in their body. If their hair is not white, when I use the word white, I don't mean the psychosocial definition of white, the way we use white here. If you're not black, you're white, or something like that. I'm talking about white like this paper white, okay? And your hair is white because you have no pigment. So if your hair is anything but white, and your eyes, the reason your eyes are red is because it's the blood, the color from the blood behind the eyes. Because you realize, do you ever take a picture of someone? And with a flash at night especially, and you'll see red eye? That's especially animals, because animals have larger pupils. Their openings are bigger, and so you get more refraction and reflection from the back of the eye. That's the light from your flash hitting the back of the eye, where there are blood vessels, and what gets emitted back out to your lens is that red color. So when you see red in the eye, that redness is blood, right? Does that make sense? That blood absorbs every other color except for red. Okay. So an albino person has no color to their eye. I have hazel eyes or brown eyes. Somebody in here might have blue eyes. Those are colors. An albino person has no pigment to their eye. And that's a recessive trait. It's a single gene that's the master gene for all the pigment in your body. Is that clear? But skin color, other than albinoism, skin color is actually controlled by 8 genes. There is no black gene or white gene. There's no such thing. That's why race is not a scientific term. Race is a term that's developed by people to try to understand the differences among us. It's not scientific. It's the opposite of that. And if it's distracting, and it's actually a lie. Race doesn't exist. Cultures exist. African American culture, for instance, Hungarian, British, Russian, Puerto Rican, whatever. Those things exist. Those are cultures. But race doesn't exist. There's no such thing as white people and black people. They don't exist. There are 8 genes. 8 genes that control your skin tone. And that's just skin tone. Your eye color is controlled, I think, by 6 genes. And so every gene has an influence on a piece of one trait, which in part explains how 10,000 genes can control millions of traits. Because it's not just oh, I get little a and I'm white, and I get big A and I'm black. No, that's not how it goes. That also explains how two black people can have a white baby and how two white people can have a black baby without anyone cheating. It's possible because black and white is not a thing. It's 8 genes that combine in a very specific way to come up with a skin tone. That skin tone could be light or dark or in between, and that's it. Yeah.
So how do people's eyes change color?
You know what? We're getting in a lot. We're way off base. Let's get back to what's important. What is going to be on the test, okay? But it's a good question, and I love your questions. I got to say, I wish we did not have an end of course exam, because if we didn't, we could spend time really investigating some things we all would love to talk about. But let's try to focus on this. We have 22 chromosomes. We call them autosomes. These are autosomal chromosomes, right? They're body chromosomes. They don't control what? What do you think if I say body, what's the other option? Sex. So that there's 2 sex chromosomes. There's one that looks like this, and then there's a little tiny, little nothing chromosome. It's actually getting smaller. We think that the Y chromosome is getting smaller. It's called the Y chromosome. The X chromosome has many, many, many genes on it. The Y does not. Every other chromosome in the body, you need 2 copies of it to function normally. If you only have 1 copy, you got problems. If you got 3 copies, you got problems. You don't want any more or less than 2 chromosomes for every other chromosome in your body. Number 23, you only need 1. One of these. You only need 1 X. Only need 1 X. If your Y doesn't work, you're going to be all right. You'll be a female. Even though you have a Y chromosome, if it doesn't work, you still got, all you need is 1 working X chromosome. Females have 2 X's. What do you think they do with it? They can't have both working. If you have both working, you have too much protein being made. What do you think they do?
Get rid of one.
Good decision. Yeah, it's not right, but they don't. Remember, we don't generally get rid of chromosomes or what we do is we turn it off. Did you say that? I didn't hear you. What do you want me to say? I can't hear you. It's not my fault. You turn it off. You turn it off. You just like you turn off a switch. It's chemically done, right? It's not like your cell says, oh, I guess which switch I'll turn this off. But you chemically shut it off, and we could talk about that after school if you like on a detail. It's called epigenetics. It's methylation, adding methyl groups to the histones, wrapping things up. There are regions that don't code, not genes, right? That's part of the DNA that's not genes. That's what it does. It regulates, it turns genes on or off. Yeah.
So you give it back to the baby once you turn it on once the baby comes?
No, actually, a baby gets it. Baby gets 2 X chromosomes. Then randomly, as you start to do mitosis, in one cell, 1 X is turned off. In the other cell, the other X is turned off. So randomly, one of the 2 X's is turned off. Yeah.
So like once we evolve, and completely understand develop and completely understand genetics, sooner or later we can just program our babies pretty much if we understand it.
Wow. Yeah, we're already doing that. In China, they've already started to program babies. Yeah. Yeah. No, it's not that they're more advanced. We could do it here. I can't really do it, but I could work on towards doing it. You could do it. We could do it here. The world has chosen to say no.
It's unnatural.
It's not because honestly, a lot of people will say, here's the thing. Watch this movie called Gattaca if you want. It's a really good movie. I'll give you extra credit if you watch it. I think it's an amazing movie. Gattaca. Get this. This is how you spell it. You ready? Gattaca. Why? Think about it. Gattaca. DNA. Right? Gattaca.
I don't know.
Huh?
Like black like programming it, like changing the gender?
Yeah, you can program, we can to this day. The hard part is turning a specific chromosome on or off. That's what we were talking about. That we can program cells. I could take your baby, and you might say, you can use something called CRISPR, and I can change the genes on your baby's chromosome. I could take and make your baby have blue eyes. It's your baby, but you're like, I don't like my brown eyes, I want blue eyes. I want my baby to have blue eyes. Okay, boom. Now, the danger is who chooses what's good and what's bad?
That's right. That's a
Who chooses that? Why is blonde and blue-eyed our choice? Why is black hair or brown dark skin our choice? If we in the movie Gattaca, you'll see it's a really interesting movie. It's a Hollywood movie. It's very good, well produced. I'm willing to show it at the end of the year, but I can't show it now because we have to get going. But if you watch it, we can have a nice discussion on it. It's very interesting. It's called Gattaca. But in any case, one chromosome, you only need one of these two chromosomes to actually get anything done. If you want to be a male, you want to have testicles and be all that a man or boy or man is, you need a Y chromosome. And the genes are on the Y chromosome. That's all Y does. It just makes the male. In the womb, we're all female. All of us have vaginas. We all have labias. We're females in the womb. As we develop, as boys develop, our Y chromosomes turn on. And our testicles form, our various pieces and parts start to form, and we turn into boys. But we all start out as girls. So if our Y chromosome never works, we just stay a girl. Does that make sense? Yeah.
Sorry I keep asking a lot of questions.
No, I don't mind. I just want to try to get moving on the content. The only concern. Go ahead.
But if you said the Y is starting to leave, so you're saying it's not going to be any more males, or it's going to be a...
Oh, I see. Yeah. And what we know is evolutionarily, we've looked at the Y and it's getting smaller. It's a very tiny little piece of nothing, really. It just has a few genes on it. That's one of the reasons we have sex-linked traits. When we talk about sex-linked traits, we usually are talking about X-linked traits because the X chromosome is very important. It has a lot of genes on it. It's a very important gene. Very important chromosome. It's very big, has a lot of genes on it. The Y chromosome only has a few. It's only important to make boys. Not just that, but it only has a few genes. There are a couple diseases that happen because of a gene on a Y chromosome, but not that many. Yeah.
So if the Y chromosome is getting smaller, would that mean the population will...
We don't know what it means. And to answer your question, we don't know what it means. It's just an observation. It's getting smaller. Some people are concerned that we don't know what that means. We just don't know. It could. There are examples of species that have evolved themselves into a dead end. I'm not sure what that means for humans. And remember, we do have technologies. Yeah. Yeah.
I'm not sure whether or not this is true, but I read somewhere that if you take a woman's bone marrow and you insert it into another woman, it can also conceive a child, not just...
Girl, code.
Okay.
All right, let's stop. We're getting into science fiction now. We have look. It's just clear. To have a child, you need 2 haploid cells, and we'll talk in a minute why that's important. All right, but let's just leave it at that. You need 2 haploid cells. Can you take 2 sperm and make a baby? Yeah, but you still need an egg. Because you remember what a cell is. What's a cell? Yeah, a eukaryotic cell. A human cell has what? Structure. It has what? Organelles, right? Where does all that come from? When you were at one point you were one single cell. What did your father give you? What did your father contribute to who you are?
Another one.
Just DNA. Your mom gave you your Golgi body, your endoplasmic reticulum, your mitochondria, and the other half the DNA. So in fact, mitochondrial DNA is a way we trace your mother's lineage. We can follow your mother's all the way back to Eve.
What?
We go all the way back. We can follow the DNA in your mitochondria all the way back, generation to generation, all the way back to what we call mitochondrial Eve, the first human female. We can do that because every one of our chromosomes comes from our mothers. The rest of our chromosomes, did it come from our great-grandfather or great-grandmother? I don't know. There's no way of telling the difference. But your mitochondria you only get from mom because that's what's in the egg. The egg is everything you need. The egg is the house. The dad is just giving you the DNA, the other half of who you are, as far as your genetic code is concerned. Yeah?
So you know how you said a child needs 2 haploid cells. So when they get those, they become a different.
That's right. That's when it becomes a viable human being. Before that, you're a sperm or an egg, you can't develop into a human being. You only have half the DNA. Remember, how many do you need? You need 2 of each of these, right? And a haploid only has 1. In your ovaries, ladies, you have 10,000 eggs waiting. They're in stasis. Every month, one of those eggs starts to develop, and whichever one gets to the outside of the ovary first, it pops almost like a zit, and very quickly it rushes out of there at a very fast pace down your fallopian tube and towards your uterus. And if everything's right, it'll meet some sperm and you'll have a baby. Those two will come together and you'll have a diploid cell and you'll have a baby.
What is it called when you have twins?
It's another story. Well, let's not talk about that right now, but it's a good question. Write it down. Yeah?
You know how you need 2 haploid cells, right? So can a diploid cell ever become a haploid cell?
It does. Yeah, that's right. That's how you make eggs, and that's how you make sperm. All your eggs and sperm came from a diploid cell, came from you. Then you stimulated that process of a diploid becoming a haploid is called meiosis. All right, and we're gonna study that hopefully tomorrow or after the test. All right, we ready? So sex chromosomes. Your X chromosome is 2 of them make you a female. An X and a Y make you a male, but understand you only need 1 X. One of the 2 X's is going to get shut down. It has to, or you're going to get sick. You're going to be sick. You can't have both X's making all their genes. You can have an X and a Y, and perhaps that's why the Y is so small, right? Because you don't want it doesn't need to be producing all those other things. All right, but in any case. This is a very special case, these 2 chromosomes, because number 1, there's only 1 of each of them, right? And even if you have 2 of them, even if you have 2 X's, 1 of them is going to be shut down. All the rest of these, you need 2 chromosomes to function normally. The other thing is that, what does that mean? Who determines sex? Ultimately, who determines sex? The male or the female? The male. Why?
Because they're going to either be one of the X or they're going to be.
They're going to be an X or a Y. If they give an X, it's a female. If they give a Y, it's a male. They determine ultimately who gets if you're male or female. Now it turns out it's a little more complicated than that. The female does have an influence on whether the child is male or female because the female produces certain liquids in her vagina, in her vaginal canal, that are either mucousy, thick, or thin. If they're thick, it favors the sperm that has the Y. If they're thin, it favors the sperm that has the X, or something like that. So there's some influence by the female on whether it's a male or female. The fetus is male or female. But overall, at a very basic level, it's the male that determines if it's a boy or girl. Which makes the whole Henry VIII thing so much sadder.
Right, that's what I was about to say. So I'm like, so it wasn't.
It wasn't the poor Anne Boleyn really didn't have anything to do with it. It was really poor Henry that was making all those girls, and he was stupid, and he killed his wife because it wasn't her fault. But his wife suffered, died, divorced. There was a whole new religion created because this guy really thought that it was the woman's fault that he had all these girls, when it really was his fault. If you want to say fault. I don't know why we say fault, right? But that in itself is a kind of a problem, yeah.
So the whole, I don't know what it is, but, it's just a myth where it's like, if you eat watermelon, there's, a higher chance.
No, all that's nonsense. No. Can environmental factors influence whether you get a boy or girl? It's possible, but we don't understand it. Again, what did I just say? The thickness of the fluid could influence boy or girl, but you're only talking about increasing the chance of getting a boy or girl. And really, if you have enough babies, you can have one, a boy or a girl, right? Unless there's something wrong with the Y chromosome, and then every time you start to produce a male, you have a miscarriage. Does that make sense? So there's all kinds of possibilities. All right, so that's everything. That's where we're going to stop for the test tomorrow. Review it. I'll post this video if you need to review. It'll be a short test, but it will be a test. It's your first test of the second semester. Please make sure you've uploaded all your documents. And yes.
And they're like kind of programmed that way, and if you lose them, it would like have to like start over again.
Yeah, but that happens all the time. Yeah. Yeah.
I was saying maybe stomach cells,
Why stomach cells?
because you needed to process the food through your.
That's right. You need all your cells, right? If you don't have a cell, if you don't need it, you don't have it. But you replace them. You replace them all the time. You replace cells all the time. Your skin cells you need, but you replace them. Your stomach cells you need, but you replace them. Not as often as your skin cells, but often. Why brain? Why? What is it that brains do that's so difficult would make it not advantageous to change them? Yeah.
The basic motor functions in the brain, it's very hard and complicated to get that down, and also in click with your nervous system, so I figured it would probably be harder to connect them all right back together again.
That's an amazingly brilliant answer. That's exactly right. Yeah, that's one big thing. There's another big thing. So I mean, you got to think about it. Your brain is a million or so. How knows? I don't even know how many nerve cells leaving your body. You're from your brain. You're wired up, aren't you? You're sending little neurons from your spinal cord out to every muscle of your body, including your face, controlling everything that you do. Each one of those has a specific path that you learned, and very difficultly, by the way, you learned how to control them. When you were a baby, it took you about a month to figure out how to get your body to turn on its belly, right? It took you another couple months to figure out how to crawl, and then a few months after that, you were maybe wobbling and falling. You probably knew how to cry almost right away, but it might have taken a little bit of time even to learn how to do that. Because remember, you weren't even breathing before you were born, not with your lungs anyways. So you have learned how to control your body over time, and your nerves have very specific paths. It is very difficult, which is why if someone becomes paralyzed, it becomes very difficult to reconnect them. Why rarely do they on their own get better, start walking again. Once you're paralyzed, it happens, but it's very difficult to get someone to walk again on their own. Does that make sense? You have to retrain all your bodies, regrow all those paths. That's very true, very good. Much more complicated than the simpler answer I was looking for. What is it that you know that lets you know that you have these cells for your brain cells your entire life? Do you have any old people in your life? Yeah, I see her. Do you have old people in your lives? Like me? Parents or grandparents? Oh, not as old as me? That's cute. Anybody? Yeah?
Is it because we like think our whole lives? So
Very close. Yeah, you think your whole lives, but you can always replace a thinking cell, right? So this thing makes a decision one way or the other. I can replace that cell. What's the thing you can't, if you lose it, it's gone, yeah.
Memories?
Memories. I remember my mama back when I was little. Some of the earliest memories. That's 50 years. That cell has been around for 50 years.
But some memories eventually die off.
They do die off, and they can get transferred over to another cell. But your nerve cells are there, and one example of that is that your memories are there. Those memories are they're not all stored. Like my mother is not stored in a neuron, right? But there are qualities of that memory that is in the specific neuron. And you're right, you do lose nerve cells your whole life. You start to lose them. Poof, poof, poof. Drink alcohol, bam, kill them, right? Take some drugs, boom, That's it. You get hit in the head, play football or soccer, you hit your head, boom, go to a few more neurons. So you lose them, but you never replace them, or not as often as you, or not as well as you lose them, not as easily as you lose them. Yeah.
Okay, so why does that happen? Like what she just said, like you lose some of your like those memories or whatever. Like you can remember something that happened 50 years ago, but like you can't remember something that happened last week.
That has to do with the nature of memory. There's something called short-term memory and long-term memory. And this goes back to study and why so many of you have trouble with these exams. There's a difference between understanding what I'm saying to you right now and remembering this in 2 weeks or 3 weeks or a month. And the trick is that you have to repeat it 3 or 4 or 5 times. You have to use it in order to get it, to put it in the long-term memory. You have to make connections between this thing that I'm saying to you now, whatever it is, and the rest of your experiences. The more connections you make with the memory, the longer you'll keep that memory. So if you want to remember something to do well on exams and you think it's important, then you need to start making connections between that information and all the other experiences you've had in your life. And that includes, that's why they tell you if you meet someone and their name's Cammie, right? That's Cammie. And they're your new boss, or they're the wife of your new boss or something, and you need to remember that woman's name's Cammie. You might say, Cammie the camel.
Wow.
Camel. So by thinking of something that's funny, or thinking of something that connecting that name to something that you already have in your brain, you're making a long-term connection. You're remembering these things. Also, some deeper experiences, more meaningful experiences tend to last in your brain longer as well. The other thing to consider is that memory can be impacted by stress. So that's part of the reason why witness testimony is the worst kind of evidence in the trial. People don't remember correctly. It's against our nature to do so. That's not how memory works. Memory is not a videotape. Memory is not a phone recording. That's why that's why I've been trying to tell you for months now, take notes. Because how you remember is not, what you remember is not what happened. Almost guaranteed. It's how close it is to what happened. Depends on a lot of different things. But it's almost guaranteed that what you remember is not what happened. Yeah.
So does that mean like people with photographic memory could like have a mutation or something?
Yeah, that does not exist. There is no such thing as photographic memory. There are people that remember very well. And they have really good ability to do that. They give something else up. Everything in this world is a give and take. You can't have it all. There is no Superman. There are autistic people, autistic savants, they can look out a window and paint the most beautiful painting, exactly what's out there. And they can do it quickly. If you've ever seen, you can go look it up and watch documentaries. There are some savants that can tell you when you were born. You give them a phone number, they'll tell you where it is, who lived there, who had that phone for the last 15 years. They'll tell you everything there is to know about people. They're like a giant database. They can't tie their shoe. They don't know your name. They can't feed themselves, but they can do all the other stuff. There's always a trade-off. An average person, we've evolved to be able to do all the things we need to do. The extra stuff, we have to learn how to do it. We have to go out of our way to do it. So rarely should you be jealous of a genius, because with genius comes other burdens that you don't know about. Yeah?
This Krishna said, can you borrow your threads?
My what?
Spool of thread.
I don't have any thread. All right. How long does a cell live? It depends on the function of the cell. Skin cells are replaced consistently, yes? And they have to be, because it's the nature of skin. Skin is constantly being rubbed, constantly being scratched, constantly needing to be replaced. So they have to be able to continuously do mitosis. Remember, mitosis is the division of the nucleus. They have to constantly do cell division, right? When is DNA copied? That's a question we're going to answer in just a second. When is DNA copied? When do you copy DNA? Yeah?
We need to replace cells.
When-
Like if you break the bone, the skin cells.
That's right. When you need to replace cells. So keep that in mind. When else do you need to?
When you have a baby.
When a baby? What about the baby? What does the baby do?
They latch on to the inside of the baby.
What do they do? What did you do? Grow. You grew. When you grow, your cells don't get bigger. It's not like each one of your cells got bigger. You made more of your cells. Yeah.
The DNA, whenever it needs to be copied, doesn't it get copied in metaphase?
We'll talk about that in a minute. And no, it doesn't. Yes.
When cells like get too old, don't they?
That's right. When they get old, when they die, and they need to be replaced. That's exactly right. Yeah. Yeah, some cells get old and they die. They just can't handle it. Also, think about liver cells. Think about what the liver does. One of the functions of the liver is to clean any toxins. You eat something that's poisonous, the first thing it does, your liver destroys it. If it can, it detoxifies it. Alcohol, for instance, there's an enzyme that breaks alcohol down in your liver. This is why your liver is your first line of defense, which is why people that drink too much end up with cirrhosis or damage in the liver, because the liver is overtaxed. And even though this liver can replace itself, can do cell division and replace a lot of its cells, there's a limit to what it can do. So when you abuse that ability, you lose the function of that liver. Yeah.
I got a question. So you said there's enzymes in your liver that break down alcohol. What if you never like drank alcohol in your life? Do they just sit there?
Enzymes, your body has regulatory mechanisms. And so as you need alcohol, your body produces what you need to get rid of that alcohol. If you drink alcohol and you have the gene for it, you start to produce alcohol dehydrogenase. Without that enzyme, if you don't drink alcohol, you're not producing a lot of it. If you drink more of it, it's just like eating sugar. I have a certain amount of insulin in my blood. If I eat a candy bar, my body starts to produce more insulin. There's a signaling mechanism that tells my pancreas to start producing insulin if I eat sugar. If I don't eat sugar and I'm starving, it signals my body to release glucagon, which is another signaling protein that tells my body release sugar into my blood. Does that make sense? If everything's working normally. So you have a bunch of signaling mechanisms that go back and forth that turn the genes on or leave them off. You have like this what's called a background production of the protein, just a little bit all the time. But if you drink a lot, then you start to produce a lot of it. Yeah.
Okay, so I don't know if this is true or whatever, but like I saw something. It was like this man, every time he ate something that had like sugar in it or something, his alcohol level would go up.
Yeah, it's possible. It's possible. Alcohol is in a sense, it's a type of sugar. So alcohol is actually full of calories, and that's one of the reasons why bacteria and yeast can make alcohol, right? So it easily takes glucose and change it into alcohol because it's a form. It's just a small change to make an alcohol from a sugar. So yeah, it can happen. Yeah.
So like the liver disease when you like overwork your liver.
Cirrhosis.
Cirrhosis, is that like the equivalent to like diabetes, but
No.
like for a different organ?
A good question. Diabetes is a lack of function. Your pancreas starts to lose its ability to make insulin. The liver is actually losing cells. You actually put holes in your cells, in your liver. You're destroying your liver when you have cirrhosis, and it's much more aggressive problem.
How does diabetes happen? You overwork your pancreas?
Your cells become what's called insulin resistant. All right, so your cells start to not respond to the insulin because there's so much insulin all the time, and the exact mechanism I can't tell you, But all that insulin all the time starts to impact your ability to respond to the insulin. And then the cells of the pancreas that make the insulin also start to get exhausted and stop working. So you get a mixture of both things happening. And it turns out that there's probably a genetic component to this. You're probably predisposed to it to some extent. We see diabetes in every culture, but what we see is there's a higher degree of a higher percentage of people in Hispanic and African American cultures that have diabetes. Now, is that because of the environment, or is that because of inheriting a specific type of gene? We're not sure. We haven't, to my knowledge, there's no gene that has been found for type 2 diabetes. Type 1 diabetes is inherited. You get it and you're not able to produce insulin at all. So that's why those poor kids, they start to take injections right away.
I was saying, doesn't DNA replication happen when they take one of your cells and they put it in a lab and then they make more of it, like if you were burned?
That's right. It does do that. But let's go on. You're absolutely right. That is one thing they do. That's one way. When you go into a burn unit and you've been burned, they'll take skin drafts from your leg or from areas where you haven't been burned, and they'll grow it on a plate. That's cell division. That's skin growing. That's what skin does, but we can encourage it to do it more. Right? When is DNA transcribed? We're going to look at that. And what must happen if DNA is to be copied? 1, and 2, if you're going to get 2 identical cells. Keep that in mind. You want 2 identical cells in the end, if we're talking about mitosis. Let's look at the life cycle of a cell. This is in general the life cycle of a cell. How long each of these phases takes depends on the cell. We start in G1. There is a thing called G0. G0 is kind of a pause. Hey, some people, you might think of brain cells as being kind of in G0. Here in G1, when you're in G1, you're actively getting ready to divide. If you're going to make 2 identical cells in the end, you have to make 2 of everything, right? Does that make sense? It's not just copying the DNA. You have to make copies of everything. Your Golgi apparatus, your endoplasmic reticulum, all your ribosomes, your mitochondria, your cytoskeleton. You have to have a double everything. Otherwise, you're going to have one cell that's not going to have enough. Yes? Mm-hmm. I mean, I think it's common sense. I mean, I think it's pretty straightforward logic. Do you agree? Mm-hmm. All right, so G1 is the copying of everything except for DNA. That's what's happening. This you need to know by next Friday, okay? This cell cycle. The S phase is known as synthesis. What do you think is happening at synthesis? You're creating what? DNA. Copies of the DNA. This is what's happening. I saw your hand up. Copies of the DNA. So this is DNA replication. It'd be nice if they called it R phase for replication, but synthesis is also creating, and you're creating copies is what you're doing. So this is where DNA is duplicated. You're making copies of the chromosome, not just the DNA, but all the histones. And we'll talk a little bit about histones in a minute. And anything else that's happening is being copied right here during S phase. G2 is when you're double checking the various things that you just copied. Do we have enough Golgi body? Do we have enough endoplasmic reticulum? Do you have enough DNA? Is the DNA correct? And any mistakes? Do we have too many chromosomes? Too few chromosomes? What's going on? So you have all these checks and double checks that are going on in G2 to make sure you're ready. Because once you start M phase, you don't want any mistakes happening. Because when mistakes happen in mitosis, that's when you get things like cancer, right? This is the life cycle of the cell. A cancer cell is just one of your cells that does this. Instead of doing this, it goes much, much faster and doesn't stop. There are signals here that tell your cell, relax, we don't need to divide, we're good. But when the when the mechanism breaks down, the cell is eternal. Cancer, you know, you say vampires, and everybody talks about vampires living forever and all that. Cancer is forever. Cancer cells are eternal. They until you die, then they die too. But they are immortal cells. They're cells that that don't stop doing DNA replication, don't stop doing cell division. Yeah.
Is there gonna be a video, or do you want us to copy this down?
You're gonna, you should copy this down, but it's also in your reading. When you do your reading for chapter 10, I believe there's a cell cycle in there somewhere.
How did we read chapter 12 that day?
I like going over the DNA structure before we do the chromosome. That's just my preference. To me, it makes more sense. So I did 12 before we do 10 and 11. I don't like, I personally don't like talking about a chromosome when you don't know what DNA is. Yeah?
So for G2, that's the DNA polymerase mostly checked, because I think that's what.
I'm sorry, say that again?
So the DNA polymerase, is that what checks the DNA to make sure that it's
What a great question. DNA polymerase is making the DNA here, the copies here, but it's also checking for mistakes here. So that's when DNA polymerase is involved. Over here, there's a double check going on. There's all kinds of different enzymes that are getting involved in making sure you have this right number of chromosomes and everything's in its right place. Okay, I don't want to get into details on what those things are. I don't know most of it, but some of it that I do know, I don't want to talk about. Because it's just going to make it more difficult to get the idea. Yes? All right, so G2 is checking that, but a very good question. All right, so now once you've done everything, and there's checkpoints, right? There's a checkpoint here, so someone just mentioned DNA. If there's a mistake here, and there's a checkpoint, I believe, here, and there's a checkpoint here, so it's actually, or maybe just right before. At these checkpoints, if there's something wrong, the cell dies. The cell dies. If there's a mistake, just like remember we talked about miscarrying, that most pregnancies or most pregnancies, conceptions end up in miscarriages because of mistakes that might be happening, and the body just rejects it, and that's it. It's over, right? Well, during cell division, that's what's happening here. The cell's going through, checking and double checking. If there's something fundamentally wrong, the cell dies. It's called apoptosis. It's over. When you have a cell that is cancerous, those checkpoints are not, there's something wrong with the checkpoints. They keep going. They keep doing this cycle over and over and over again. And you get, that's what a tumor is, right? Instead of having a regular brain tissue, you have this giant lump because the cells keep doing cell division and they keep growing. And then they start to impede on the function of everything else that's going on in that particular area of your body. So mitosis is also known as M phase. This says mitosis, but you can replace, the reason we call it M phase, why would you think we call it M phase? Yeah? Okay, yes, but I can see that, but it's not just mitosis. What might happen in the same area? Somebody, yeah, meiosis. It could be mitosis or meiosis. Either one can happen here. But in the case of every cell in your body, every autosome, every body cell in your body, the only thing that's happening is mitosis. You're just making copies. Mitosis is taking and separating, dividing the nucleus. That's what mitosis is. Are we clear on that? Mitosis is the dividing of the nucleus. Cell division is here. What is that word? What word do you read? Cytokinesis. Kinesis is movement, like kinetic, right? Movement, kinetic, yes? All right, and cyto means cell. So cell division is cytokinesis. That's what happens. That's it. That's the cell division. This is the life cycle of the cell. Mitosis is just the dividing of the nucleus, dividing up the DNA. Are we good?
Yep.
All right.
So what?
Yeah.
Kinesis means dividing.
Kinesis means moving, right? Kinesis. Mitosis, cytokinesis means dividing of the cell. That's the actual division of the cell. We'll talk more about, you'll read about it this weekend, hopefully, and I will talk more about it next week. So here's this cell cycle again, drawn out for you. There are various phases. And let's talk about what those phases are. So interphase is everything except for mitosis and cytokinesis. So that whole G1 and G0 and S and G2, that's what we call interphase. Right? That's all the stuff that's happening outside of mitosis. Interphase, the phase in between mitotic events. So mitosis and meiosis are going to happen here, and this area at the end of interphase, you start to prepare for mitosis. This is where you start to see chromosomes. Chromosomes look like this. They're condensed around histones. When they're not, when it's doing all the protein synthesis, the G0, G1, G2, G1 or G2 or S phase, everything's opened up. The DNA's unraveled. We call that chromatin. There's still a DNA molecule. Remember, how big is a DNA molecule? Like 3 meters long, right? It's kind of crazy. But a chromosome looks something like this. There's two of them, right? Because you've made copies. This is only after synthesis, only after DNA replication. You have 2 copies. They're exact copies. We call these sister chromatids. All right, they're just exact copies. Now remember, what is that? That's just DNA, 5' to 3', double-stranded. That's it. Except that it's so long, in order to move them efficiently from one side of the cell to the other, you have to pack them, right? Does that make sense? You're not gonna, if you're gonna move to a new house, you're just not gonna pick up your sock drawer, all the socks in your sock drawer, and run out the house with them, right? You're gonna put them in a box or a bag or something. So you got to take this 3-meter long DNA, and you're gonna have to wrap it around these proteins, and we call these proteins histones. They're actually divided into 4 little pieces, and we sort of like hair curlers, right? You've seen women with hair curlers before? You guys use hair curlers? I don't know if your generation does or not. Flexi rods or whatever you call them. You take your hair and you wrap it around something to give it a curl, right? Well, you take this DNA and you wrap it around these histones, and what you end up with after you wrap it around all these histones, you end up with something that looks like this. You end up with this thing here. It's just a nice, compact, wrapped up piece of DNA. Each one of these is a chromosome. Remember, chromosome is still, when it's unraveled, when it's not wrapped up, we have another word for it. Unfortunately, you have to learn all of them. It's called chromatin. So chromatin is when the DNA is unwrapped. Chromatids, sister chromatids, is the 2 copies after synthesis. And a chromosome is when everything is the DNA all wrapped up.
You said 2 copies after what?
Say what again?
You said the sister chromatids were the 2 copies after what?
After synthesis, after DNA replication. Does that make sense to everybody? So this is a chromosome right here. Do you see this? This is a chromosome. 2 copies of the DNA wrapped up around histones, connected together by a centromere. That's what this word is here. That's the name of this thing here, is it? Centromere.
The thing in the middle?
That thing in the middle, yeah. Centromere. All right, you'll learn more about it as you go on. Please remember, one more thing before you go. When you separate, when these two sister chromatids get separated, they will also be known, each one will be a chromosome again. So when these get separated, there'll be 2 chromosomes. So it's a little tricky. All right, good luck reading. We'll talk more about this Monday. How'd you do on the test, by the way?
I don't know, I didn't check.
I got a question.
Yeah, a lot
Wait, did you upload it?
Yeah.
90, 70, 90, 90, 60, 70, 60, 60, 50, 50, 80, 80, 80, 90, 50, 80, 80, 90, 100, 100, 70, 60, 90, 80, 50, ask them, 70.
So nobody got under.
You'll know them when I put them in the grade book next period.
Wait,
I just told you this class scores. I think that I don't think that's that bad.
The other class be thinking they saying they had 30. Are we your best biology class?
No,
Who's your best biology class?
I'm not sure. What do you want me to say? Like by grades?
Yes. It's definitely it's our class. What? So
You're tired. I'm just going by grade. You want me to go by average grades, right? You want the highest scores of each class? Or do you want the average grade of each class?
Right, like your
The best performance by class average is 6 period. They beat you by 0.5% second quarter. You're both about the same. Not 6, I'm sorry, 4th. 0.5, you're both about, I think you guys had an average of 85%, 85.2, and they had 85.8 or something like that.
look at this.
I'm lying. Let's take a look. I don't know. You asked that question. I don't know.
Look at this.
What is it?
That's you.
What do you want me to do about it?
I was like 0.5% away.
It's your problem. You should have thought of that when you missed assignments.
I didn't miss any assignments. You can look. I did all
First
my work.
period had 89%. Third period had. Yeah, I was right. Oh, that's third period. This is the advisory. 82%.
Ours was 89.
Yeah, so far it's you. Then 86. 6 periods 86.
We win.
Well, let's see.
We win.
Hold on, we have to look at the second period.
We win.
84. Yeah, you guys are the best, but not by a great deal, anyway. It's not my fault. You should have done harder.
I tried to.
Work harder this time. I don't know what to tell you.
You should give me some extra credit.
No, I already did. Look at it.
You should give me more.
No. You have an exam today? It's going to take your whole period. The packet will be due next week. It's on the, please, you're going to need every minute you can.
Are you going to post our work on the computer? Or that's on one day?
That's not stress, you dieted. It's not stress. That's why I was like, oh my God, this poor kid's seriously worried. Like, turn the lights on.
That's on Monday. You said we have an assignment Monday, Tuesday, and Wednesday.
They're all in the grade book. It's out of your email. Yeah?
So what I was saying, I said the packet, right, that you said to do.
Not today.
I know. The answer's on the back.
Good. I do that. I don't care. I want you to do it. We'll see who understands it, though, today. Somebody just came to me and said, if you don't leave, I'm writing you up. I'm writing a referral right now if you don't get out. I am not playing with you. Once you come in this room. All right, so you have an exam today. It is. Can I tell? I have this in there actually. At least you know when he gets older, he'll look good.
You got the wax?
Yeah.
That's the hair wax?
Huh?
Oh, never mind.
They ain't trying to.
Why is everybody so
No, because
fixated by
she
it?
would get her hair done every other day.
Why you happy?
She was like, she do all the techniques. She was like, you got the wax.
Yeah, I still say you two would make a nice couple.
I'm talking to.
Man, those,
You
you
two.
guys gotta put somebody on the spot.
I'm just saying, you two would. I think if you take her, I bet you ask her out to movies, she'd say yeah.
I bet you I won't do it.
That's what they say. I'm just saying. I'm just saying.
Man, you trying to.
And I'm playing Cupid. Right.
I ain't playing Cupid. I'm just making an observation, and I'm.
You trying to play God.
Girl look good with Johnny. Everybody look good with Johnny.
Well, he's a nice guy.
Everybody.
You know, my nephew has just graduated from college, and he's rarely goes out with. I mean, he's very shy. It reminds me of Johnny in many
He's
ways.
never shy.
And I don't know. Like you, he pretends and he fronts, but with like. I don't know. It just bothers me. Youth is wasted on the young. That's all I have to say. Youth is wasted on. Youth is wasted on the young.
I'm like R. Kelly.
If I could have your youth and know what I know, I'd rule the world.
12.
All right. All right, guys, come on.
What is this over with?
You're not gonna be happy with your. I don't want to hear you running out of time. You're not listening to me.
Ooh, Tristan. I thought you was whipping.
All right, number one, if I catch a cell phone, I'm looking for cheaters today. So I can't wait to catch somebody. Honestly, I don't want to sound like I'm a vindictive individual, but I honestly cannot wait to catch somebody cheating. I'm going to enjoy myself that day. When that day, and the day is coming. I don't know when, I don't know where, but it's coming. I don't know if it'll be today. It might be. If it is, that day will be a day of joy and celebration.
Are we gonna be laughing?
I know. I said I don't want to sound vindictive. The reason I said that is because I know it is vindictive. All right, so. Ow, I just hurt myself. So I looked at the multiple choice test, and I don't think you're ready for it, so I'm not giving you a multiple choice test today. Today, you'll get it next week, Wednesday.
Oh, okay, that's cool.
I want to give me some time to go over some of the stuff that's on that test that I don't. Can I finish? Could you listen? And these tests that I'm giving you, the multiple choice tests, are all secured tests, so I know you don't have access to the questions. And they're secured tests. They're secured by an organization called NIMSY. Those are people that make me go all over the daggone country for stupid meetings that I'm tired of.
You don't want to travel at night.
Man, you don't know. See, again, youth is wasted on the youth. So, anyways, you have no idea what it's like to travel. It's a pain in the butt. They grope you, they frisk you. Your stuff is all in a little box. They sit you next to some stinky person. You got to sit in a freaking airplane for a couple of hours. And
For free?
yeah, whatever. It is for free, but it's a.
You need to be happy. Go get a window seat.
First of all, let me be clear. It's not for free. I have to put $500 up front. They pay me back, so that's true.
Okay, but you get it refunded.
But it's still $500 out of my pocket that I can't use for me. And it's every day I'm out, I do double the work when I get back.
If you don't like being a teacher, don't do it.
All right, I'm done talking with you about this. You get me started. Don't get me started.
If you paying up front.
And staying up at a hotel away from my wife. The only thing I want to do is hang out with my dogs and be with my wife. No, it can't. There's too many.
I know, I feel you now. Bro.
You're taking the test, whether you like it or not. And she's right. You need the time. You're taking it today. It is an essay exam. It's an FRQ. You're not listening. The multiple choice you're not ready
You
for.
passed me that out here already. I got a sharpened pencil.
Good. All right, so you're gonna need some paper, some clean blank paper.
I got y'all. I found a notebook in here.
Can I get one?
And you need to separate yourselves in the room, and then I'll hand out your, it will take you all period to do this FRQ. It's a 1,000-point exam,
Why you not like a bubble something?
because it's a full FRQ, full 2010 FRQ.
Based off of this? Oh, it's based off of this?
No, that's they stuff.
It is pollution, yes, sir.
So he's talking about the
All right. So it's not gonna be easy, but I think if you use your heads, if you use your heads and you put your time in, and of course it's a standardized answer, so the answers are already written. I will grade it based on the AP. God, why do you keep talking, man? You guys. So it's based on the 2010 free response questions. That's what you have. Yes, let me finish. If you think about it and you read the question carefully and you can write on the test itself, so that's not a problem, but you're gonna need some blank paper, and you need no notebooks out, just 4 or 5 sheets of blank paper. It's a full-length FRQ. You go ahead, let's keep talking, that's a good idea, let's do that. There's 4 questions, and there's about 4 sections for each question. Make sure, I'm grading it like I would if I was grading the AP exam. So make sure that you label answer 1, and then 1A, and then 1AI, and then 1AII, and 1AIII. Because every question is broken up into sections. Make sure you're clearly stating what the sections are. I will only grade the first answer if I ask for one answer, if the question asks for one answer, I'm only grading the first answer you put down. Don't put 5 things down and hope that they get one of them right.
What you mean by that?
Okay, I'm saying it again. I'm grading it based on what the test graders have told me they do. All right, the people that grade this test have told me what they're told to do. When they grade, they look at the answers. If they ask you for one answer, list one source of pollution, blah, blah, blah. And you put 5 answers, and the first one's wrong, and the last one's right. The question is wrong. I'm not reading after the first one. If the first one's right, and the last 4 are wrong, doesn't matter. The first one was right. You got that question right. So it does not help you. In fact, it might hurt. It can't hurt you either. But you want to put your best answer first. Is that clear? Whatever else you do. Make sure you list break it down, circle if they ask you for a number and you're doing all kinds of calculations, you must do it by hand. So there's no, am I still recording? My goodness.